Hello everyone, I welcome you back to another important lecture. And for today, we would do the topical past paper practice for one of the technical topic that you have got for your IGCSE Biology, and that goes with the title of Human Nutrition. And we would do the questions all the way from the year 2016, and we would go till the end of 2025. Now, if we were to look on toward the first question that came in the May-June of 2016, the past paper variant for your theory was 4.2. And in the exam, this was question number 2 with you. And they say is that this figure 2.1, we show you an electron micrograph of the bacteria, and the bacteria is Vibrio cholerae. What we know is that the bacteria, as they form their own kingdom of the prokaryotes, and among that, they have shown you the electron micrograph of this Vibrio cholerae bacteria. And if we were to go to what the examiner has to be asking you was that for many years ago, the scientists that we have with us, so they discovered that this bacteria that they have shown you above, this releases a toxin. And they look on toward the effect of this toxin, and then they are presenting you the results of their experiment, and they were trying to see that how come this toxin that is released out by the bacteria, this can impact the flow of the chloride ions out of the human cells when there is toxin and when there is no toxin. Now, the idea usually here is that in elementary canal, what we have got is the small intestine, and in the small intestine, we have these finger-like projections. And these finger-like projections, they are the villi with us, and within the villi, we have this human cell, right? And this human cell is what we call this as the epithelial cell. The usual idea is that if you have eaten some contaminated food or contaminated water, it would have the bacteria, Vibrio cholerae in it, and when this would release the toxin out of it, so the result of this toxin would be that it would compel these epithelial cells to be uh, releasing more of the chloride ions into the intestinal lumen. And what the intestinal lumen is, that basically is the space or the location that we have got within here, within the small intestine. So if the toxin is there, so out of the villi, more chloride ions would come here. And we know as in that impact, more water would get out of these epithelial cells and the person would suffer from the diarrhea. Now, if we just look on toward the graph, the first point is that we need to look on the key. The key is that the graph indicated with the crosses. This is when there is presence of the toxins, and one that is with the dots. So this is without the toxins. What we can be seeing here on the x-axis is this is time given in seconds, and on the y-axis we have got the flow of the chloride ions in terms of the arbitrary unit. And I can simply see that when there is presence of the toxin, so the flow of the chloride ions is greater compared to when there is no presence of the chloride ions. In the graph, you see on the x-axis, small 5 units that we have here, they are equivalent to 2 units. So this would mean that each small unit on the y-axis, this is equivalent to 0.4 arbitrary unit. Likewise, if we come on toward the x-axis, so in terms of the time, from 50 to 100 seconds, each small box that we have, that would be representative of 10 seconds with us. Now, if we were to just go on toward the first part that they ask for the 2 marks is that we are required to be calculating the difference that is there in the flow of the chloride ions between the cells when they have the exposure of the toxins and the cells when they don't have the exposure of the toxins, and that too at 50 seconds. Now, what we need to be doing at this point is that we would come on toward the graph for looking for the 50 seconds. So here you see that I have got this 50 seconds. Now here we have the flow of the chloride, and we need to be calculating the difference in the flow of the chloride ions. when there are toxins here in this graph and when there are no toxins. So from 50, I come here toward the curve for no toxins. And when I go on toward the y-axis, so here the value that we are getting is 0.4. Likewise, we would go further here, and what I see is that here the flow of the chloride ions in the presence of the toxin is 12 arbitrary units. So since I have to be calculating the difference in the flow of the chloride ion. So now I have got my value. So what I'm required to be doing is that this value, I would subtract it from this value, and then we would have our difference in the flow of the chloride ion. So as I have written here that when the time is 50 seconds, so when there are toxins, so with toxins, the flow of the chloride ion is 12, and when there are no toxins, so the flow of the chloride ion is 0.4. So we simply need to take the difference among it. So 12 minus 0.4, this is giving me 11.6 arbitrary unit. If we were to go on toward the second part, for 3 marks, they say that you are required to use the data that is presented to you in figure 2.2, and you need to describe the effect of the toxin on the flow of the chloride ions out of the cells. So in description, we just need to be elaborating the graphs. There has to be no logical answer, or there has to be no explanation coming here. So if I were to just take you back to the graph, and if we were to be simply describing it, so what I see is that for the graph, when there is presence of the toxins, the flow of the chloride ion increases and then decreases. So this is the first general point that we need to be writing in the description. The second point is that you look for the maximum point. And what I see is that when the time is 50 seconds, so the flow of the chloride ion is maximum, and that is 12 arbitrary units. So this is the second description point. What I see next is that from the time 50 to this much time, and between 100 and 150 here, the time would be 125 seconds. So between 50 to 125 seconds, there is rapid decrease in the flow of the chloride ions. And how much that would be? You would say that it would decrease from the 12 arbitrary unit, and at 125 second, this would come to 4.5. So here we have come across three description points. I am going to tell you the fourth description point that you could be writing as an alternative point. So what I see is that between this time, which is 150 seconds, to 250 seconds, the flow of the chloride ion increases and decreases, and then slightly increases and decreases. So for here, what you could be writing is that between 150 to 250 seconds, the flow of the chloride ions remains relatively constant, or you could say that in between this time, the flow of the chloride ion fluctuates. So this is how you were required to be writing it in terms of the description. So for 3 marks, how the answer would look like as in accordance with the mark scheme. So it would be that there is rapid increase in the flow of the chloride ions during the first 50 seconds, as I've shown you above, and it peaked to 12 arbitrary units. And then I indicated as above that this is decreasing to 5 arbitrary unit, and that time was 125 seconds. And then for the time that was here from 150 to 250 seconds, so we would say that here the flow of the chloride ion remains fairly constant, or you could say that it fluctuates. So this is how you would have scored total of 3 marks for this. And if I were to move to this third uh, part of the question, so they say is that chloride ions cannot move out of the cells by simple diffusion. Okay, that means that if I were to have this epithelial cell and it's there in the villi of the small intestine, so if we have got the chloride ions within them, so they cannot go out of these cells by simple diffusion. This is what they have told you. Now they're asking for your suggestion, and they want you to describe that how the chloride ions could move out of the cells. So here the point is that wherever there is a hint to the presence of ions, and we know ions are the charged particle. So they would always move out of the cells or into the cells with the help of the active transport, because we don't have any other choice. Because for the water, this is osmosis. And if the particles they move from the higher to the lower concentration, and if they are the not charged particle, it would be simple diffusion. But here in this case, since the ions are the chloride ions, which are charged particles, so we would say that these ions would now move out of these cells with the help of the active transport. And we know it very well that whenever active transport is involved, so there has to be the releasing out of the energy from the mitochondria that would do it with the help of the aerobic respiration. And once this energy is released, then these ions they would be moving out of the cells with the help of the transport proteins. The general aspect is that the ions, since they cannot cross the cell membranes directly, rather than they would always be in the need of these transport proteins. So this is how you would score total of 3 marks for this. If I were to move to the D part, they say is that the loss of the chloride ions from the cells causes diarrhea and dehydration in the patient with cholera. Okay, fine. We are convinced the patient has got the bacteria in his body, and once there would be the deposition of the chloride ions within the lumen of the small intestine. So, for sure, here in the lumen, the water potential would be less, and ultimately, out of the intestinal cells, the water would come out here into the lumen, and that would bring about the dehydration and the diarrhea. The thing was, they wanted you to state which organ in the alimentary canal is affected by the cholera toxin. So this is very simple. That organ is the small intestine. Actually, this is the target site where the Vibrio cholerae bacteria would come and would be exhibiting its effect in terms of the diarrhea. For 2 marks, they want you to describe the treatment of the cholera. Now, considering the fact that cholera is a bacteria, so this means that we can be using the antibiotics that would either kill the bacteria or would stop the reproduction of the bacteria, or their divisions would be stopped. And in the diarrhea, we lose a lot of salt and ions out of our body. So the other treatment protocol would be that we can be having this oral rehydration therapy. And you may have heard that in the market there comes some sachets that actually have got the mixture of some sugars and the salts and the ions in it, and we call this as ORS. You usually go into the market and purchase it. So that is the oral rehydration therapy. And since this has salts and ions and sugars in it, so once you would have it, ultimately it would compensate for the loss of the ions that were previously lost in your diarrhea. Moving onward, here we have got another question, and here the examiner would like to assess that how good you are with identifying the parts of the alimentary canal. So this appeared to be in May, June of 2016. The theory variant was 4.3, and this was question number 6 for your exam. What they tell you is that we are giving you this figure 6.1, and it shows you the alimentary canal of two mammals. One among them is an insect-eating bat, and that insect-eating bat is a carnivore. And then the other diagram which they have presented you, that is of the rabbit. And they say is that this rabbit is a herbivore, meaning that it is feeding on the plants. And the insect-eating bat, since it's carnivorous, so it would be feeding more on toward the meat stuff. So if you were to look on figure 6.1, here they have given you the insect eating bat, and on my right hand side, they have given you the rabbit. What we can see as clear identification that we see is that this here is the esophagus, and this is the esophagus. This and this is the stomach. And immediately after the stomach, like irrespective of the parts of the small intestine as duodenum, jejunum, and ileum. So this part is the small intestines. And likewise, if you were to be talking about the insect-eating bat, so after the stomach, this part again would be in the small intestine with you. And then Here, this part in case of the rabbit is the large intestine, inclusive of the cecum, colon, rectum, and then finally here we would come toward the end of the alimentary canal, that would be the anus with us. They ask you is that you are required to be naming the organ that is labeled V and W, okay? So V that we have here, so this is the stomach, and W that we have got here, so this would be the large intestines with us, this part. So for 2 marks, V is the stomach and W that we are given here. So this is the large intestines with us. Now here for 3 marks, what the examiner wanted was that you explain to him the role of the mechanical digestion. What we are familiar is that within the alimentary canal, the digestion could be the mechanical or it could be the chemical digestion. Wherever the mechanical digestion is involved, so there would be no enzymes doing the breakdown of the food, but rather than some physical processes would be involved. And wherever there is a mechanical or the physical digestion, so what we know is that the large food particles that we have with us, so they would be broken down into the small particles physically, like by the action of the teeth that we have in our mouth and by the action of the muscles that we have in the stomach. And when that would be a situation, that these food particles, when they are broken down into the small pieces, so their surface area is increased. And when their surface area is increased, then it makes easier for the enzymes to be breaking them down more efficiently. So how your answer need to look like for the 3 marks in context of the mechanical digestion should be that this mechanical digestion, this breaks down what? The large food substances into small pieces. Here you would get 1 mark. And then we are telling him that this is done with the help of teeth or with the help of muscles. Like this is your choice whether you write teeth or the muscles. And you would say that when they come into the play, so that would increase the surface area of the particles. And when the surface area of the particles is increased, then it becomes easier for the enzymes to be effectively working on these small particles. Now here you would score 3 marks. As an alternative approach, you would also say that the role of the mechanical digestion is also that it can be allowing for the easier swallowing of the food. And you can also say that in mechanical digestion, the food is not chemically changed, but rather than this is physically changed in terms of a reduction in the size of the particle. If I were to move toward this part that we are given in terms of figure 6.2, let us see what the examiner has to be telling us about this graph. What the scientists did that they got different species of the mammals, and in these different species of the mammal, they are investigating the digestion. And the animals which the scientists they studied, they had a difference in their size. Like it ranged from very small mammals, that was an elephant shrew, which is Elephantulus edwardi, and you see it has a very little mass of the 50 gram, and comparative to that, what they have got in their investigation was an ox. The genus of that was Bos, and the species was Taurus, and it had a mass of 220 kilograms. So you see is that their investigation is involving the animals of very small mass to a very bigger or a heavier mass at in case of the ox species. Now, how they performed the investigation was that these scientists they took some indigestible particles and then they added them to the animal's food. And then what they measured was that how long these particles they would stay in the digestive system. They took or they carried this investigation for a total of 24 different mammal species, and then the results they are showing you in figure 6.2. Now, if we just come on towards figure 6.2, so here on the x-axis we have got the relative body mass in terms of the arbitrary units, and then on the y-axis we have got the time for how long these indigestible food particles it was staying in their digestive system, and that in terms of the hours that we have. Now, before I go with cracking the question or what the examiner had to ask from you, what we see is that as the relative body mass is increasing, the stay of the food or the indigestible particle in the alimentary canal, this is also increasing. Okay, this is also increasing, but what we see is that these 2 points that we are given here, so they don't go with the usual trend, which is followed by these points. So for such data, we could say that this is actually the outliers, or they might be some anomalous result that can occur due to some error in their investigation or due to some error which the humans can make when they were required to be doing the calculation at the end of the investigation. Let us see what the examiner has to be asking us in terms of the question. So for the total of 4 marks, what the examiner is telling you is that the scientists they concluded that food stays longer in the digestive system of larger mammals compared with the smaller mammals. So this is their conclusion. Like if the animal has a bigger mass, so the food would stay longer in their digestive system. And if we have the mammals with a small mass, then the food would stay for a short period of time within their alimentary canals. They want you to discuss the evidence that too from figure 6.2, and you need to go in favor of it and against the statement that the examiner has presented here that food stays longer in the digestive system of the larger mammals. Since the marks are 4, so two we would be writing in favor of it, and two statements we have to be writing against their statements. So I would not read this point, rather than we would go back to the curve and we would see that how we are required to be writing the answer as such. So you see the first point here should be a very general point, and what I see is that as a relative body mass of the animal increases, the time for the food to stay in their alimentary canal also increases. Now, this is the general point in favor. Now, I need to be quoting him my favor of their statement in terms of some numeric justification. So, if I just see at this body masses that is smaller body masses, and if I were to just hint him to this point here, and that is lying in between 1.5 to 2.0. So here the relative body mass would be about 1.75 kgs. And if we just try to see that if their body mass is around 1.75 kgs, then how longer the food is going to stay. So each small box here is representative of 4 units. So this is 4, this is 8, this is 12, this is 16. So this is coming halfway. So I would say that at a body mass of smaller body mass of 1.75 kgs, the food stay is smaller, and that is 14 hours. Likewise, the other point would be in terms of a heavier body mass. So let me just like I'm not including these because they are the outliers. So rather than I would talk with respect to this point. Now, this is the highest time that we have got in terms of a bigger body mass. So you see again this point that I have with me. So this is coming in between 5.0 to 5.5. So this would be 5.25 body mass. So, at this much body mass, if we see that the stay of the food is more in terms of the hours, and if I were to just highlight it from here, and we are coming some halfway here, so you see again each small box was 4 units. So this is 84, this is 88, this is 92, this is 96. So this would be 94 hours. So these statements they are going in the favor, as with the smaller relative body mass, the food of stays smaller. With a heavier body mass, the stay of the food is longer. Now, this is in favor of the statement which the investigator they presented. Now, in against, what I would tell him that the data which they have presented here, the outliers, they do not follow the general trend. And for this, I would say that the result for the two species is anomalous, and we can never be sure about the anomalous result. The other point in against would be that the sample size is small, and when the sample size is small, then the data we don't rely much on it. So this is how the answer was required to be written by you. Now, whatsoever I have highlighted in the graph, so I'm not going to be speaking it up for you. So we would say that as the body mass increases, the time for which the food particle stays, that too in the digestive tract this increases. Then I have told him the same point that when the body mass is about 1.75 kgs, the food stay is smaller for 14 hours. But where we had a higher body mass of 5.25 kgs, the food was staying for the total of 94 hours. So these are going in favor. Now, again, I would go against it. So I would say that when we have the body masses that were a bit smaller, like 3.4 around to 3.8, the time for the two species is anomalous, or you can be writing an alternative point that the outlier shows a relative higher stay hours, even though their body mass was smaller. And then against the last point would be that the sample size is small and which is only limited to 24 species only. So I hope this would make sense now that how we were required to be writing it in favor or against the statement. Moving on to the next question, and here the examiner was interested to evaluate your understanding of the enzymes that contribute their role in the digestion of the complex food particles into small food particles within your alimentary canal. Appeared to be in October, November of 2016, and the theory variant was 4.1, and in your exam, this was question number 3. What they have to be telling us is that pepsin, this is a protease enzyme which is found in the alimentary canal. For 1 mark, the examiner wants you to tell him the product which would be formed from the digestion of the protein by the protease enzyme. And the answer would be very simple. As we know, the proteins, they are made up of the amino acid to be making the polypeptide chains, and they further make the protein. So if the protein would be acted upon by the protease enzyme, so the final product that we be getting, this would be the amino acids with us. For 1 mark, they say state the organ in the alimentary canal where the pepsin is created. And this is a fact that pepsin is released in the stomach, which is a major part of your alimentary canal. Now, the B part that we have here with us, so this is a kind of investigation which they were performing. What the biologists did, that they performed experiment. And the motive of that experiment was that uh, the motive of the investigation was that they wanted to find the optimum pH that is for the activity of this pepsin enzyme. And we are aware of this fact that the activity of the enzyme is affected by the temperature, by the amount of substrate, by the amount of the enzyme, and by the pH. So they want to look on toward the optimum pH for the activity of this pepsin enzyme. How they performed the experiment was that these biologists, they took total of 4 test tubes, and in each of the test tubes what they added was 1 cm cube of cooked egg white, and that cooked egg white has proteins in it. In this figure 3.1, they say that we are showing you these 4 test tubes, and you see that since we are checking the optimum pH, so the pH we would be changing. So this is our independent variable. As you see, all the 4 test tubes have a different pH, so this is the independent variable. Now, afterward, what the biologists they are measuring is the time taken for the complete digestion of the cubes of the cooked egg white. Like they would simply see that in the test tube 1, 2, 3, and 4 having different pH, in what pH the cooked egg white would be digested in less time, and that would be their optimum pH. Now, for 2 marks, what they were telling us is that the biologists ensured that all the cubes of the cooked egg white were exactly the same size. And they want your suggestion that why that was so. Now, as I have said, that the activity of the enzyme is impacted by the temperature, by the pH, by the amount or the size of the substrate and amount or the concentration of the enzymes. And in this investigation, we were interested to find the optimum pH, and the pH we were changing. So this means that the other factor like the temperature or the surface area or the amount of the enzyme or the substrate they would change, then we would not be able to see the effect of the pH alone. So you could say as that why all the cubes were of the same size, because this would keep the surface area of all the cubes same, because we know that the enzyme's activity is affected by the surface area. Or if you don't want to be writing this point, so as an alternative bullet point, you could say that it would allow them for a better comparison. For 2 marks, what the examiner is saying that the temperature must be controlled in this experiment. And they say that describe how the temperature could be controlled. We are aware of this fact that if the temperature is very low, then the enzymatic activity would be very slow. If the temperature goes very high, so the enzymes would be denatured. So that is why we are required to be controlling the temperature. But they wanted you to tell him that how you would control the temperature. So there are a lot of approaches to be controlling the temperature, like either we are required to be keeping the water bath in an incubator or in a thermostatically controlled water bath. Other approach would be that we would wrap up the test tube in which we are doing our experiment with the insulating material. And then again, we could say that we can be using a thermometer to get an idea of what temperature would be there in the test tubes, and that would help us to be controlling the temperature. For the C part, they say that the same experiment was performed with trypsin, and that is another protease enzyme. In the above, they were talking about the pepsin, that is a protease enzyme, but in the stomach. But they say that trypsin is another protease enzyme, and that works actually in the small intestines. And the pH of the small intestine is alkaline. So, since they wanted you to predict the optimum pH of trypsin, and considering this is acting in the small intestine, so we need to write the values of the alkaline range. And in this case, it is in range between 8 to 9. If I were to move to the D part, so they are relating this concept of your enzymes in the digestive system or the alimentary canal in context of the enzymes. And for the 6 marks, what they were to be asking is that enzymes such as protease, they are important in digestion. They want you to describe in detail that how the enzymes they function using other digestive enzymes as an example. So they gave you the name of two enzymes. One is pepsin, the other is trypsin. Other than that, we need to quote him the enzymes that would be working in our digestive system as digestive enzymes. Since the marks are 6, we need to be describing to our examiner the total of 6 points. So how the answer should go as like we would initially tell him that the enzymes they are made up of the protein, because the first part of the question is in terms of how the enzymes function, and then we have to quote him some examples. So we would say that the enzymes are made up of the protein, and then we would say that they have the shape of their active site, and that is complementary to the substrate that would fix here and would make the enzyme substrate complex. Afterward, we would say that this, in case of the digestive tract, the digestive enzyme would break down this complex food substances into the small molecules and hydrolytically, because most of the chemical reaction that goes on during the digestion of the complex food particles, they are hydrolytic reaction. They occur in the presence of the water. Then we need to quote him some examples, and we would say him that the enzymes like the amylase that break down the starch into glucose, or we would say that the lipase that break down the lipids into fatty acid and cholesterol. And at the same time, we would say something like that during these reactions, the pH, the temperature, They has to be maintained. If we are not be controlling them, then the enzyme would be denatured. So this is how you would score a total of 6 marks for this. Now coming up to this question, and this appeared in the October-November of 2016, and the theory paper variant was 4-2, for your exam, this was question number 1. A mega question this was in that year, total of 21 marks. And here the examiner was interested to know your evaluation in terms of how good you are to be telling him in terms of assimilation, that once the digested food particles are absorbed and when they become the part of the cells, how good you are with it. And then it would be related to the enzymatic activity in your alimentary canal, and there was some part that we would see in a while that was related to the lactose intolerance situation, where we are not able to break down the lactose into glucose and galactose. What they are telling us that milk is sometimes referred to as a complete food because it contains all the nutrients that a young mammal requires. And in the table, they have shown you three nutrients that are available in the milk, and they want you to state one role of each nutrient in the body of the young mammal. So it is related to, as I said, to assimilation and to the biological molecules. The protein that we have, this is a fact that it can be used for growth, or either you could say that it can be utilized for the repairing of the damaged tissues. Like one from these 2 points. The lactose, overall, we know has got glucose and galactose in it, and once it would be broken down, so we would get glucose out of it for the respiration. For the calcium, we know that this strengthens bone, or you could say the teeth. And this is also required for the muscles contraction. So out of these points, you would require to be writing any one. If we come toward its B part, and that was for the total of 6 marks, they say that protein digestion begins in the stomach of the human alimentary canal and is completed in the small intestine. Whatsoever the protein we would be eating in the stomach, the pepsin would break it down into the polypeptide chains, and when that are carried into the small intestine, then the trypsin enzyme would break it down into the amino acids. The first part of the question says that describe in detail how the enzymes function. And then the second part is that you are required to relate it to the protein digestion and that too in the alimentary canal. So the first 3 points they should be there in terms of the enzymes function, general point, and then the other 3 points they should be in terms of the enzymes responsible for the protein digestion. So either we would write it in context of pepsin or trypsin. So the first three general points are that the enzymes are made up of the protein, and then they have their shape of the active site, and that would be the complementary to the substrate molecule. And when the substrate would collide with this enzyme's active site, so they would make the enzyme substrate complex. So here you would score a total of 3 marks for the first part of the question. And now let us become specific to the protein digestion. Now, either you would say that we have the pepsin enzyme in stomach, or if you don't want to write it in terms of pepsin, you could be writing in terms of the trypsin, but you have to relate him that that is in the small intestine. No matter if it's pepsin in the stomach or trypsin in the small intestine, they break down the protein hydrolytically in the presence of water to their respective amino acids. And at the same time, we have to be telling them that for the enzymes responsible for the protein digestion, the pH has to be maintained. The pepsin enzyme that we have in the stomach, this requires the acidic pH of 1 to 3, and the trypsin would require the alkaline pH of 7 to 9. At the same time, the last point would be that the optimum temperature of 38 degrees centigrade is also required because it would bring about the effective collision and would save the enzymes from denaturation. So this is how you would have cracked total of 6 marks for this part of the question. Moving on to word, its C part, and that is relative to the lactose intolerant people. If there is any person and if this is lactose intolerant, so they do not have the lactase enzyme in their small intestine. And without the lactase enzyme, they are not able to break down the lactose, which is the milk sugar, into glucose and galactose, and that is why they suffer from the lactose intolerance. They have given you figure 1.1, and here they are trying to give you an idea that what would happen if this lactose intolerant person would be eating up the food or be having the food in the diet that had got a lot of lactose in it. So if you were to just follow this diagram, from the stomach, the lactose is entering into the small intestine, and since they do not have the lactase enzyme, and the lactose being a double sugar would not be able to pass the cell membranes or the cells of the villi of the small intestine. So now this has come into the large intestine. In the large intestine, we have got the bacteria, and these bacteria they would break down the lactose into the simple sugars. And once the simple sugars are made, so the bacteria would use them and they would produce the hydrogen gas, the methane gas, and the carbon dioxide gas out of it. As a result, these persons they would build up a lot of water and the gas in their large intestine, and they would suffer from diarrhea and a lot of the gas passing out of their alimentary canal. Now let us see what the examiner has to be asking you. For the first 2 marks, the examiner asks you that you are required to explain why the lactose is not absorbed by the small intestine. The idea is that this lactose itself is a double sugar, and if we do not have the lactase enzyme, so it would not be broken down into the simple sugars like the glucose and the galactose, and that is why it would not be able to cross the cell surface membranes and it cannot be absorbed by the small intestines. So, how the answer should look like? You would say that lactase enzyme is not available to break it down into the glucose and galactose, and lactose sense as a double sugar and is large enough. So, alone itself or without being broken down, it cannot be able to cross the cell membranes. If I were to come toward its second uh, question for the total of 3 marks, they say suggest the dangers to the health of severe diarrhea if it is not treated for a longer time. So you see, when the person has the severe diarrhea, so the ultimate outcome of this would be dehydration. And why dehydration? Because in diarrhea, the person would lose a lot of water from its body, and there would be loss of some essential mineral ions like the calcium ions would be lost. In order to score the third mark, we are required to be telling him that what negative impact would be if there is loss of the ions like calcium ions out of the body. So the calcium ions are generally required for the proper nerve impulse conduction by the neurons, and when there would be loss of the calcium ions, so the electrical impulse transmission by the neurons would be highly affected. If you forget about any of this point, you could also be writing something as that if we are losing a lot of water in the diarrhea, so as a consequence of that, the volume of the blood plasma would decrease. And when the volume of the blood plasma would decrease, then the cells would start to lose the water, they would shrink, and in severe cases, if we do not compensate for the loss of water and ions, this can also kill a person. Moving on toward the D part, this is related to the diagram that they showed us above. And here the examiner is presenting you the way where we can be monitoring the lactose intolerance. And for what they are saying is that, okay, fine, in the small intestine, the lactose was not digested, and when it came into the large intestine, so over there, the hydrogen gas was produced. This hydrogen gas, it would be getting absorbed into your blood and then it would be excreted through the lungs. So for the lactose intolerant people, a lot of hydrogen gas they would be exhaling out. And By measuring this hydrogen gas content that is exhaled out or breathed out, we can see that how much is the severity of the lactose intolerant person. Now, they added some people into the investigation and then they gave them different milk products. For the people, those who were there in the group A, they were given the untreated milk, like that would have the pure lactose in it. There were the people, those who were given the milk, but that milk was treated with the lactase enzyme immediately before drinking. For the third group of the people, they gave them the milk, but they added the lactose into it 3 days prior before the people they drink it. And then the D group, it had included the yogurt, and this yogurt was made by the bacteria. And the good thing about the bacteria was that it is able to digest the lactose in the milk. Now, after these people they were given these kind of the milk product on different days, what the scientists they measured later was that how much amount of the hydrogen gas was breathed out by the four group of the people as A, B, C, and D, and they measured it for every hour for the total of 5 hours after they ingested each of the milk product. Now, if we were to just come on toward the graph, and I have already labeled them up, so you give me an idea that what protocol among them is the most feasible one. On the x-axis, we have got the time in context of the hours, and on the y-axis, we have got the hydrogen gas that is breathed out in terms of parts per million. What I see is that the people, those who ingested the untreated milk, the hydrogen gas production in them is the max. Those who were drinking the milk but was treated with the lactase immediately before they drank it, so the amount of the hydrogen gas released out is has decreased comparative to A. For the C people that had the milk but was treated with the lactase enzyme 3 days before, again, the amount of hydrogen gas is less than B and less than A for this much time, but afterward this is exceeding beyond the people, those who were in the group C. But if I just see it for the D people, D group people, so in them, the hydrogen gas production is the least as at 2 hours, the maximum hydrogen gas produced is this much, like that is nearly 15 parts per million. And overall, this has stayed the lowest from this time onward comparative to the other groups. Now, for the first part, for the total of 2 marks, they said that why untreated milk was included in the investigation. Now, since we know that in the rest B, C, and D, there is treatment for the lactose, but for the A, the milk remained highly untreated. So, such kind of a setting, the experimental setting, this acts as a control always. And why we include a control in our investigation is so that we can make a valid comparison with the other treatments. For the next part, for the total of two, they say why lactase might be added to the milk. We know that the lactose intolerant people, they do not have the lactase enzyme in it. So when we would add the lactose to the milk, so it would be broken down into the glucose and lactose. By that, they are small enough, they would be able to pass through the small intestines into the blood, and ultimately the bacteria that previously were making the hydrogen gas, the carbon dioxide, and the methane gas in the large intestine, so they would not be able to do so. So they would now not suffer from the diarrhea or having producing a lot of gas in their large intestines. For the third point, and the marks were three, they said that you are required to use the results that are present within figures 5.1.2 to explain why yogurt is the best milk product for the people with the lactose intolerance. So in order to crack 3 points to score 3 marks, let us revert back to the graph and see that why yogurt is the best milk product for the lactose intolerance people. What I see is that comparative to the rest of the treatment protocol, for the yogurt scenario, the amount of the hydrogen gas released is the minimum, right? Like if I just talk in context of the time at 5 hours, so the hydrogen gas production, this comes to be 10 parts per million. And even for the rest of the time, like from this time onward, comparative to the other groups, the amount of hydrogen gas produced has remained less, and it is also showing less of the fluctuations comparative to the rest of the groups. So the answer would simply look like that we have got the lowest amount of the hydrogen gas being released, and then we are quoting him that this was 10 parts per million at 5 hours, and comparative to the other milk treatment groups, this was comparatively the lowest. Also, this shows the lower fluctuations than the rest of it, and the concentration of the hydrogen gas at any cost never exceeds beyond 15 parts per million. Like you see, like the max it reached was 15 parts per million, and it's not exceeding more than that. If we just see for the other groups, the hydrogen gas released is more in it. So that is why this yogurt treatment protocol is more useful than the rest of it. So see, this was such a mega question, and any of the student who has not studied it, he would have lost 21 marks for their exams if they were appearing in that year. So let us just now come to the past paper variant 4.3, and this was for your October November 2016, and it appeared to be question number 3 of your exam. Most context of this question is in terms of the absorption, and with a slight question being asked in terms of the role of the bile in the human alimentary canal. What the examiner has to be telling you is that we have got four type of the mammals, and in those four type of the mammals, we have measured the length of their small intestines. And their results they are shown in figure 3.1. And if you were to see the mammals given are four, we have got insect-eating bat, we have domestic cat, we have the rat, and then we have got the human coming with us. For the second column, they have given you the length of the small intestine in terms of the centimeter, and we could simply see that the smallest length is for the insect-eating bat. The largest length of the small intestine is for the human, and it is set at 552 centimeter. The third column is in terms of the length of the small intestines, that is relative to the body mass, and that is in terms of centimeter per grams. And what we can simply be seeing here is that as the length of the small intestines increases, so the length of the small intestine relative to the body mass decreases. So that indicates a kind of a negative relation or the negative correlation between the length of the small intestines relative to the body mass. For the first A point, for the total of three, they ask is that you are required to use the information in figure 3.1, where they ask you to compare the length of the small intestines of the four. And this is something very simple. We simply need to make the comparison of the lengths. So what I see is that the smallest length of the small intestine is for the insect-eating bat, that is 19 centimeter. The longest small intestine is for the human, that is 552 centimeter. And if we were to see the domestic cat and the rat, so there is difference of only 6 centimeter. Like here for the domestic cat, this is 104. For the rat, this is 98. So comparatively the same, relatively the same, but with a slight difference of only 6 centimeter. So how the answer need to look like is that the length of the small intestine, this is longest for the human, which is 552 centimeter, and this is shortest for the bat, which is only 19 centimeter. And what I have said is that the domestic cat and the rat, the length of their small intestine is very similar with a slight difference of 6 centimeter. Moreover, if you were to write any alternative value point, so you would say that the length of the small intestine has a negative correlation with the body mass, because if the length is increasing, the relative body mass comparative to the relative body mass, so this decreases. So this would help you score 3 marks. Now, if we come to the B part, the total marks are 3, and they tell you is that this figure 3.1, this is a diagram which is showing a short length of the small intestines of the mammal. So they have shown you the small intestine of a mammal, and in the diagram they show you the outer surface, then they show you its inner surface, and then the empty space where the digested food would be available. So this is the lumen with us. Now what they have to be telling us is that the function of the small intestine is absorption, and they want them to answer, and they want you to answer them that describe how a molecule of glucose it passes from the lumen of the small intestine into the blood. What they mean is that here we have got the glucose in the lumen of the small intestine, and how come this would go into the blood? If I were to be recalling, so in the small intestine, what we have is a small finger-like projection, which is the villi. The first layer of the villi that I'm indicating as of now. So here we have got the epithelial layer, and in the epithelial layer, each individual cell is the epithelial cell. If I were to go beneath the epithelial cell, so here we have got the blood capillaries with us. Considering that the glucose is lying in the lumen of the small intestine, which is empty space here, so they want you to tell him that how come this would be absorbed into the blood. So the answer is very simple, that the absorption of the glucose would be under the impact of the active transport, not the diffusion. Now, these epithelial cells that we have here, they have got abundant mitochondria in them. These mitochondria, they would release energy in the form of ATP, and that would help to move the glucose molecules from the lumen of the small intestine, where they would be passing through the transport proteins here in the epithelial cells, and then it would come all the way into the blood capillaries. So, how your answer should look like, you would say that the molecule of glucose it passes with the help of active transport, where from the lumen of the small intestine and then through the cell membrane of the epithelial cells, it would come into the blood capillaries, and it would be done through the protein carriers. And for all the process which is written above, there has to be the energy utilization that would be coming from the respiration process. If I were to come here to the C part, what they are trying to be saying is that for the four mammals that they gave you above, they took their small intestines out, and for the small intestine of each individual mammal, they took the measurements of their inner and the outer surfaces, and then they showed their results in table 3.2. Now, again, the same mammals are written here, and here they have presented you the ratio of the inner surface area to the outer surface area, and they have done it for the duodenum, and they have done it for the ileum. For 1 mark, the examiner is asking that suggest which mammal has the most villi per centimeter of the small intestine, and you see that the highest value I have it for the duodenum of the small intestines of the insect-eating bat. So it would have more villi per centimeter, because as the surface area would be increasing, so more villi would be packaged in the duodenum of the insect-eating bat. Another for 3 marks, they say the duodenum is more effective than the ileum at absorption, and for that we need to use information that is available in figure 3.2 to explain why. Now, the idea is that we have got the ratios for the duodenum and the ileum. And what I see is that the ratios are higher, far more higher for the duodenum of these animal species comparative to the ileum. So this is the first mark, that the ratios are higher for the duodenum than the ileum. And what we see is that for the duodenum, the inner surface has more inner surface than the outside. So this means that in the duodenum there would be more villi packaged comparative to the ileum. So that is why the duodenum would be more effective at absorption. So for 3 mark, it would be the ratios are higher for duodenum than ileum. It also has more inner surface than the outside, and that would mean having more villi, and it would allow for more effective absorption. If I were to come toward the D part, and there are 4 marks. And here they say is that the bile is released into the small intestines from the gallbladder. But we know as that here we have the liver. The liver actually synthesizes the bile juice and the pigment, but for the short period of time, the bile would be stored here in the gallbladder, and then later it would go into the duodenum of the small intestines. They want you to outline the role of the bile. Now, what we know is that these small intestines, they are basically receiving the liquefied food from the stomach, and that liquefied food from the stomach goes with the name of chyme. The issue is that in the small intestines, the enzymes they require the alkaline pH. So that is why this bile, when this comes from the gallbladder, this would neutralize the acidic chyme. Other point is that we need to provide alkaline environment for the pancreatic lipases, and that again would be done with the help of the bile. And one of the most important concepts is in terms of the emulsification, where the large fat molecules that we are eating in the diet, when they are broken down into the small fat droplets, so this is emulsification, and this is again done with the help of the bile. The important point here is that the breakdown of the large fat molecule into the small fat droplets, this is not chemical but physical digestion. Other than that, once the bile has broken down or emulsified the fat, so it would also increase the surface area of the fat globules, so that would allow for the faster digestion of the fat by the lipase enzymes. So since this question was in terms of outline, you can write any four out of it. I have written five, but you prefer writing just four out of it. Now, here we come to the year 2017, and this was May June 2017. The theory paper variant was 4-1, and this appeared to be question number one of your exam. This is from your concept of the enzymes involved in the fat digestion, the absorption of the fat, and the role of the fats that we have in the body. For the A part, the marks were three. And what the examiner has to be telling you is that the fat is necessary component of the human diet, and they want you to state him three ways in which the human body uses the fats. Now, what we know is that our cells they have the mitochondria, and their role is to be releasing the energy by the process of respiration. Apart from the glucose, the fats that we have got in the body, they can also go to the mitochondria, and then the energy can be released in the form of ATP. So the first statement is that they are used to release energy by the respiration. The other valid statement is that it provides insulation, because the fact is that in our skin, where we have the top epidermis coming with us, then we have the dermis coming with us. Below the dermis, we have these fat tissues that have the fat stored in them, and they would not allow the heat to escape out of the body, and that is why the fats they provide the insulation. Our internal organs, they are also have got the deposition on them by the fat, and they help to cushion the organs. If you forget about any of these 3 points, the other valid points are that the fats are used to make the cell membranes and the hormones as well. And what hormone is fat based? This is the testosterone hormone that develops the male secondary characteristics. If I were to move toward this figure 1.1, here they say that we show you the pathway of the fat in the part of the alimentary canal. Here I have the esophagus. Then we have got this stomach here. And then here we have got the pancreas. They show you the liver here. We have got the gallbladder here. And you see the gallbladder and the pancreatic sections, they would go into the small intestines that we have here. If we were to be going toward the questions, so for the B, they say state the name of the enzyme that is created by the pancreas to digest the fat. And the fats, they are lipids. So for sure, the enzymes coming from the pancreas would be the lipase enzyme. For the next 1 mark, they want you to be naming the product of the chemical digestion of the fats. Now, the fat that we eat in our daily life, they have the fatty acids, three fatty acid chains linked with one glycerol molecule. So once the fat would be digested in our alimentary canals, so we would get the fatty acids and the glycerol out of it. For the third, They want you to name the liquid that is produced by the liver and stored by the organ P. The organ P, if you see here, this is the gallbladder. The liver produces the bile, and for some short period of time, this is stored here in the gallbladder. So the liquid present within the organ P or the gallbladder, this is the bile with us. And they want you to be naming the organ P, and as I have indicated above, this is the gallbladder. For the C part, the marks were 2, and they say what happens to the ingested fat at R in figure 1.1 before chemical digestion can occur. So if we revert back to the diagram, so here they want us to be telling us that what is happening at R before the fats can be chemically digested. So the idea is very simple, that the fat digestion can simply be split into two of the major steps. The first step that we have with us, this is emulsification, and then the second step that would be there in the breakdown of the fat, so that would be the enzymatic activity that we have got, and that enzymatic activity would be there by the specialized enzymes, and these enzymes, they are the lipase with us. So that means that before the chemical digestion happening here, at R the emulsification would be done. And whenever the emulsification is to be bothered about, so what we know is that these fat molecules that are complex and large, so with the help of the bile salts and pigments, they break down into small fat droplets or small fat molecules, and this is emulsification. And when the emulsification is accomplished, so these small fat molecules or the droplets, they have the greater surface area and they have the lower surface tension. And when that is a scenario, so it becomes easier for the enzymes to be effectively digesting these fat droplets into the fatty acid and glycerol. So the thing happening at R before the chemical digestion, as I have indicated above, that here emulsification would be done by the bile. And by that, the large fat molecules, they would be broken down into small fat droplets or into small fat globules, and with that their surface area would be increased, allowing for the efficient breakdown by the lipase enzyme. If I were to come to the D part, for 3 marks what they are saying is that explain how the product of the fat digestions are transported from Q to the rest of the body. Now, the scenario is that at Q we have got the breakdown product of the fat, that is the fatty acid and glycerol, and this says that how come this would be transported to the rest of the body. So what you need to be assuming here is that I have got these villi. In the villi we have this epithelial layer, and then we have got this blood capillary. The issue with the blood capillary is that it cannot transport the fat to the rest of the body, but thanks partly to the presence of the lacteal here. The word I said is lacteal here. So what we would say is that since we have the breakdown product of the fat, which would be three fatty acid chains, and we would have one glycerol molecule with us, so you see is that they would initially pass through the epithelial cells that we have in this epithelial layer. And from there, they would enter into the lymphatic vessel that we call this as the lacteal, and this lacteal has a colorless fluid in it. And the name of the colorless fluid is lymph, and that would absorb the fatty acids and the glycerol, and from there it would distribute it throughout the body. So how your answer should look like for the total of 3 marks, that these product of the fat digestion, they passes through the epithelial cells and then goes into the lymphatic vessel, and that has the lymph in it, and that would carry the fatty acid and glycerol into the blood. Now, here we come again to May June of 2017. The theory paper variant was 4.2, and this appeared to be question number 4D part of your exam. Marks are 3, and what they have to be telling you is that it is important that the children with marasmus produces enough bile. Now, we have nothing to do with marasmus, and they simply want you to describe the role of bile in the digestion of the fat. Again, the bile, we know that it breaks down large fat molecules into the small fat droplets or the globules, and this entire process is emulsification. And when the small fat globules are made, so their surface area would be increased, and it would now make it easier for the lipases enzymes to be effectively breaking them down. The other idea is that the intestines they require the alkaline medium or the alkaline pH so that the enzymes available there they can work optimally. So thanks partly to the bile that it would neutralize the stomach acid that would be present in the chyme, a liquefied form that enters into the small intestines from the stomach. So that would be neutralized with the help of this bile. For the same year, May June 2017, there was another question in your exam. This was question number 60 part. And they said that figure 6.3, it shows some human teeth that require dental treatment. For 1 mark, they wanted you to identify the type of the teeth that they are showing you in figure 6.3. So what I see is they appear to be looking some broader teeth and they are not denser like they are not denser but rather than they are thin sharpened teeth, so they indicate that these are the incisors with us. Moving onward, here we have got the question from October November 2017 and the theory paper variant this was 4-1 and this happened to be question number 1 year of exam, and this is from your elaboration of the structure of the villi. What they tell you is that we show you figure 1.1 and figure 1.2, and they are the two images of the villi. Figure 1.1, this shows the surface view of many villi which are viewed through the scanning electron microscope. So they have shown you the view of many villi when they were using the scanning electron microscope. The other figure that we have 1.2 that would be showing you the section of one villus that is viewed through the light microscope. So here we have got another villus but viewed with the help of the light microscope. Now, the identification of this structure is very important. So what I see is that overall this part that I have with me, this is villus, right? At the center, we see this part that is skew with us. So this would be the lacteal having lymph node, okay, having lymph node. And this guy right here that I see at this point, so this is a goblet cell, and the role of the goblet cell is to be producing a sticky substance that is a mucus with us. And this guy, which is indicated by the letter R, that we see at this point, you see, like we have got very thin layered structure with us. So this is a blood capillary. Thing goes on, and if you were to look like these cells that we have here, they are the epithelial cells of the villi, and these would be the tiny projections that we have on the top of the epithelial cells, and this as would be the microvilli with us. Okay, so this was elaboration of the diagram. Now, if I just take you to the questions, so for the A part, what they tell you is that the villi, they are found in the small intestines, and for 1 mark, they ask you that says the function of villi. So the function of the villi are numerous. The most prominent among one is that any of the food molecules that we have digested, if this has to be absorbed into the blood, that would be the role of the villi. Other than that, the villi also have got some enzymes within their epithelial cells, and these enzymes they are embedded within their cell membrane. So you could also be saying that the role of the villi also is the digestion of the food particles like the maltose, lactose, and the sucrose, but the prominent one is this. If you were to come on toward the B part for the total of 4, they say is that identify and describe two of the labeled component of the villus. We are required to use the latter that are given in figure 1.1 in our answer. As I have indicated above, the PE it was a goblet cell, and this releases a sticky substance that is referred to as a mucus. The Q that we had at the center, so it was a lacteal, and it has a colorless fluid that is the lymph with us. If you don't want to write about P and Q, so if I just take you back here, so the R was the blood capillary and the S were the microvilli, so you could also be indicating to them as well. So, as an alternative valid point, you would say R is a blood capillary and it is one cell thick. S is the microvilli, and since they are thin, so they would for sure reduce the distance for the diffusion process. If we were to see to the part C, they tell you is that some infections in the small intestine can cause diarrhea. Describe the effects of diarrhea on the body. So for sure, when we are having the diarrhea, so there would be loss of iron and we would have the watery feces. Other than that, the other valid points are that the diarrhea can also cause death if this leads to some kind of severe dehydration, and we also feel the stomach cramps whenever we are suffering from the diarrhea. For 1 mark, they asked you to state the treatment for the effects of the severe diarrhea. So this is oral rehydration therapy, or you could be taking antibiotics, but only when the diarrhea is caused by the Vibrio cholerae bacteria. For the D part, they say is that the blood transport nutrients, and they ask is that state the component of the blood that transport nutrients. So we know that is the yellowish liquid part of the blood, and we name this as the blood plasma. Right away, for the second part, what they tell us is that the nutrient in the blood can be used to become the part of the cell. The nutrients in the blood mean that after I have digested the food particles within the alimentary canal, and when I take them or absorb them into the blood, so for sure out of the blood capillaries, these absorbed food particles they would enter into the cells and they would become the part of the cell. So that phenomenon or the process goes with the name of assimilation. For the last third um, part of the question, for the total of 2 marks, they tell us is that the amino acids are the example of the type of nutrient transported in the blood. So the glucose and the amino acid and the vitamins and the minerals, so all of the stuff like that, they get transported around the body with the help of the blood plasma. They tell you is, they ask you is that state two examples of the large molecules which are found in the cells that are made from the amino acid. Now they want you to tell him the larger molecules that can be made from the amino acids so we can make the antibodies out of it. You can name some enzyme like as I have written DNA polymerase enzyme because this is working within the cells. The other large protein that we make within the cells out of the amino acid, this could be the hemoglobin protein with us. Likewise, if I were to come here, so this is from May, June of 2018. The theory paper variant was 4.1, and for your exam, this was question number 1B and C part only. In this question, your examiner was interested to know your elaboration of the identification of the parts of the alimentary canal and what role they are contributing in the digestive process. For 2 marks, they ask is that explain the importance of chemical digestion. Now, what we know is that whenever the digestion is physical or the chemical, the large food particles or the complex food particles, they would be broken down into the small soluble molecules. If the digestion is a chemical digestion, so there would be involvement of the enzymes. And when the enzymes would be involved, and when the large complex food molecules, by the influence of the chemical digestion, when they are broken down and they become small soluble molecules, so they are easily being absorbed into the blood or they can easily enter into the cells of the body. You were to see the C part, and we would just try to identify the parts of the alimentary canal in a while. They say figure 1.1 shows the human alimentary canal and the associated organs. The associated organs mean the accessory organs that are not directly the part of the alimentary canal but since have a role in the digestive process. So we discuss them along with the alimentary canal. They say the functions of some of these parts of the body they are given in table 1.1. Now, before I crack each and everything, so here they have given you the table that we are required to complete, and for our benefit, they have done the first row and the row for us. And they say is that function if is the storage of the feces, then the latter from it is F. And if you see this F is the rectum with us, and they have given you the name as well. So likewise, we are required to be doing the rest of it. And they say site of starch digestion. Now, as far as the starch digestion has to be bothered about, so this happens within the alimentary canal part, which is the mouth and the buccal cavity. And here it would be released with the help of the salivary amylase that comes out of these salivary glands. The other part where the digestion of the starch can happen. So this can happen within the J, which is the first part of the small intestine. That is the duodenum with us. Likewise, you can also be writing the letter E because here any starch that remains unleft from the mouth and when this is coming to the small intestines all the way from the stomach and the stuff like that. So here the pancreatic amylase comes. So the pancreatic amylase can break down the starch at J, at E, and we have got the salivary amylase being splitted at A. So that is why the latter that we have got, you can write A, you can write J, you can write E. And if I have to be writing in context of A, so I would be naming the structure as mouth. If I am writing letter J and E, so I would be saying small intestines. Then this is reabsorption of the water. Now, as far as the reabsorption of water has to be bothered about, so not only the large intestines, but the small intestines would also be absorbing the water. So either you write as H or you write it as E. If you're writing H, so you would say this is large intestine. If you're writing E, so you would say this is the small intestines with us. Likewise, some of the water can also get absorbed from here as well. No matter if you write J or you write E. In both contexts, the name of the structure should become small intestines. If you're writing H, you can simply write it as large intestine or you could write it as colon. They say excretion of the pepsin. So this is the fact that the pepsin is always secreted within the stomach. So the latter that we have here, so this is C and we don't have any other choice. So the secretion of the pepsin, the latter is C, and the name of the structure, we would not be writing it as pepsin, but rather than we would be writing this as stomach. Now they ask is that what is the site of the maltose digestion? The maltose digestion happens within the small intestine. Like you could be writing J, you could also write E. Both of these, they are the small intestines. The idea is that the maltose is broken down by the maltase enzyme, and the maltase enzyme is available within the epithelial cells of the villi, and they are available at J, they are also available at E. So in both regards, you could either write E, you could write the letter J that we have with us, and the name of the structure would be small intestine. The excretion of the bile. Now, as far as the excretion of the bile has to be bothered about, so the bile actually is produced and released out of the liver, but temporarily gets stored within the gallbladder. So, since they are saying that what brings about the excretion of the bile, so you have got the choice of the letter K because this also secretes the bile. No matter if this gets stored into the gallbladder, you could write L as well, that is a gallbladder as well. So it's your choice either you write K letter or L letter. But if K is coming here, so it would be liver. If you're writing the L letter here, so it should be the gallbladder. This is excretion of the lipase and the trypsin, and we know that the trypsin and the lipases enzymes they are being released out of the pancreas. So here we don't have the choice of any other letter. So the letter coming up would be the D for the lipase and the trypsin station, and the organ that we see here. So this is the pancreas with us. So this is how you would have scored total of 6 marks. Now here we come to the May-June of 2018. The theory paper variant was 4-2 and this was question number 1 for your exam, where the examiner wanted to see your approach relative to the structure of the teeth. What they tell us is that we have got the red pandas, the genus is Eleurus, the species is Fulgens, and human they say have similar arrangement of the teeth. They tell you is that figure 1.1 it shows a section through one teeth of a red panda, and figure 1.2 shows the side view of the lower jaw of the red panda. Now what we know from the structure of the teeth is that this A is enamel. Immediately below the enamel, this part that we have here, so this goes with the name of dentine. And this part where we have got the neurons and the supply of the blood capillary, so this would be the pulp cavity. And this part right here, so this would be the gum with us. And if I were to take you to this part, you see that here in between the gum and this, you could say the dentine that we have. So here comes to be gluing material, and that goes with the name of cement. If we were to be looking on toward the arrangement of the teeth that we have here, so C teeth that we have got, so they are the incisors and these pointed teeth. They are the canines, the ones that we have here, they are the premolars, and at the end what we have, they are the molars with us. Now this is what they have asked for the total of 3 marks, that we are required to state the name of the structures labeled E to F, both in figure 1.1 and 1.2. As I have labeled a while ago, that E that we have, this is dentine. The B that we have here, so this is the cement. The C that we have here, so they are the incisors. The D that we have are the canines. The E that we have are the premolars, and at the end, the F that we have got, they are the molars with us. So this is how you would have scored total of 3 marks. Let us come to here. This appeared in the May June of 2018. The theory paper variant was 4-3, and for your exam, this was question number 1 with us. In this question, the examiner was interested to know your evaluation or elaboration of the chemical and the physical digestion, as well as enzymes that are responsible for the breakdown of the complex food molecules into the simple ones. What they have to be telling us is that two functions of the alimentary canal are mechanical digestion and chemical digestion, and they wanted you to outline that where and how mechanical digestion occurs in the alimentary canal, and the total marks were 4 with us. Now, what we know is that in the mechanical digestion, whatsoever the large food substances we are eating in our diet, they would be broken down into the small particles, and there would be no chemical station involved, no enzyme would be involved. As far as where this is happening, so we know is that this would happen within the mouth, where we have got multiple types of the teeth. We have incisors, we have canines, we have premolars and the molars, and they grind the food, they cut the food, they tear the food, ultimately breaking them down into the smaller particles. The other aspect of where the mechanical digestion happens is within the stomach, because we know the stomach is a muscular bag and it has got stronger muscles in its wall. And when those muscles they would simultaneously contract and relax, so that again would be breaking down the large food molecules into the smaller one, and that too physically. So for 4 marks, how your answer should look like, you would say that mechanical digestion break down large food substances into the small particles without a chemical change. It happens where? In the mouth. And over there, I have written it in context of incisor teeth. You could write canines, premolars, and molars. And we know incisor teeth, they cut down the food particles. Then I have written for the 4th mark that it also takes place in the stomach, and where the food would be broken down by the muscular contractions. If I were to take you to the B part, the marks were 3, and this is your evaluation, testing your evaluation in terms of the enzymes that are responsible for the chemical digestion. They themselves says that enzymes they catalyze the reactions of the chemical digestion. They have given you table 1.1, and that is giving you the information about the chemical digestion that happens in the 3 parts of the alimentary canal. So if I were to just see on the table, the first part they have given us is the mouth, and they say that the substrate that we have got is the starch, and they're asking the name of the enzyme for it. So we know the starch is broken down by the salivary amylase. Like here, the word salivary has to be written clearly because they are talking with context to the mouth. Now, it would be acting upon on the starch, and we would have the maltose getting or producing out of it. Then they gave us a part of the alimentary canal stomach, and they say that we make the peptides there in the stomach. So we are aware of this fact that in the stomach, the enzyme working is the pepsin. And the substrate on which this would be working, it goes with the name of protein. For the last, they did not give you the part of alimentary canal, neither the enzyme, but they gave you the name of the substrate, which is the fat, and the product are fatty acid and glycerol. And my concept is that fat are only broken down in the small intestines, not in the mouth or in the stomach or in the esophagus. So either you could write small intestine, you have the flexibility of writing it as duodenum and ileum as well. And the enzyme responsible is a lipase enzyme. And lipase enzyme, you may remember it for your MCQs paper, that the lipase for the breakdown of the fat, this comes from the pancreas. If we were to go to the C part, they say the substances that are absorbed from the alimentary canal may enter the cell and becomes a part of the cell. So this entire statement is actually the definition of the assimilation process. For 1 mark, the examiner wants you to state the storage carbohydrate which is made from the glucose in the liver cells. Now, the storage form of the carbohydrate, we know it well, that this is a complex sugar, and the name is the glycogen with us. For the second point, they asked you is that state the type of the protein which is used in the immune system and that is produced from the amino acids by the lymphocyte. And we know lymphocytes are a specialized type of the white blood cells that releases a protein out of them, and that are the antibodies protein. And we know they clump the pathogen, they make the pores in the wall of the pathogen, ultimately killing them, and that would save our body from getting the harmful effect of that particular pathogen. For the last 1 mark, they say fat is produced from fatty acids and glycerol by the cells in the fatty tissues beneath the skin. In the skin, we have the top layer of epidermis, the lower layer is the dermis, and over there we have got these fatty tissues. And the role of the fatty tissues is that they would combine the fatty acid and the glycerol, and we would store the fat in these fatty tissues. For 1 mark, they want you to state one function of this layer of the fat. So, when this is in context of being deposited beneath the skin, so over there the role of the fat would be providing the thermal insulation. Now, if we look on toward the October-November 2018 paper, the theory paper variant was 4-1, and in your exam, this was B and C part of question number 5. This is related to the liver and its function. What they have to be telling us is that the liver is also involved in the processing of amino acid. For 2 marks, the examiner wants us to describe how excess amino acids are broken down. Now, this is related to a bit to the digestive system, but mostly this is in context of the excretory system. And when we talk about the role of the liver in the breakdown of the excess amino acids, so what we know is that from the small intestines, the amino acid that we have got from our diet, they would be coming to the liver with the help of the vein here. The name of the vein is hepatic portal vein. My body has a situation that it can never store the protein. It would always use the proteins what is required by the body. So we need to get rid of the excess amino acids. The idea is that the excess amino acid in them, they have got this group in them. The name of the group is the amine group. And when we try to take off or we try to be removing this amine group from the excess amino acid, so this is referred to as a deamination process. And when the deamination is done, so by that we would be able to be getting rid, we would be able to get rid of the excess amino acid, and we would make ammonia or the urea out of it. So the answer becomes very easy. to answer that the excess amino acid they are broken down by the deamination process. And in the deamination process, the amine group is removed from the excess amino acid, and then we make the ammonia or the urea out of it. For another 1 mark, they want you to state the name of the process that assembles amino acid to make the protein. Now, when the assembly of the amino acid is being done or the attachment of the amino acid is done with motive to be making the protein, so this goes with the name of protein synthesis. For the C part, the total marks are 2, and they tell us is that the liver is also involved in the processing of the toxins. What they have to be presenting the situation is that lactic acid is an example of the toxin that is produced during the vigorous exercise and then processed in the liver. What we know from the daily life is that whenever we are doing some vigorous exercise or strenuous exercise, so in order to fulfill the requirement of the energy by the muscle tissues, initially at the start of the exercise, both the aerobic and anaerobic respiration takes place. When the anaerobic respiration is happening, so we make lactic acid out of the glucose. And that lactic acid, since it's toxic and then cause muscle fatigue, so the liver has a role of removing it. So this is what they are asking is to describe how the lactic acid is processed. When we are resting after the exercise, the lactic acid that we have in the muscle, it comes into the blood plasma, and the blood plasma would take it or would bring it to the liver. Once the lactic acid has got to the liver, the lactic acid is broken down into the glucose, and the glucose that we are getting out of the lactic acid, this would then be used by the aerobic respiration, and we make carbon dioxide and water out of it. The other approach is the same lactic acid can be converted into the glycogen, and in that way, the lactic acid is getting processed within the liver. Why the liver is asked in this context is because this is an accessory organ of our alimentary canal. Coming here to again October November of 2018, and the theory paper variant was 4.2. And for your exam, this was question number 6B with us. The context of this question is to get an understanding of how good you are with the enzymes related to the digestion of the biological molecules. They say is that figure 6.1, we show you the Galapagos iguana. They give you the name of their genus and the species that we need not to bother much about it. If you go toward the questions, before that, let us see what the examiner has to be telling us is that Galapagos iguanas, they feed on seaweed, which contains starch and other carbohydrates. So this is the food they are eating, the seaweeds. And the nutritional content of the seaweeds is that it has starch in it, and it also has got some other carbohydrates in it. For 1 mark, they want you to state the name of the enzyme that digests starch. So this is the fact that the enzyme for the starch breakdown is amylase. For another 2 marks, they want you to state the names of 2 parts of the alimentary canal where the starch is digested. So initially, the starch digestion happens in the mouth where it would be partially broken down to the maltose. Some starch remains undigested in the mouth and it would not be digested in the stomach, but when the undigested starch comes into the small intestines, then over there the pancreas would be sending the pancreatic amylase to be breaking down the remaining starch to the maltose. Let us come here. This is from October November 2018. The theory paper variant was 4.3, and this was question number 6 from your exam. What they have to be telling us is that this figure 6.1 that we show you, this is a diagram and it shows some body cells and parts of the human lymphatic and circulatory system. So what we see here is that on one side we have got the arteriole, on the other end we have got the venuole. Between the arteriole and the venuole, we have got this part, and these would be the blood capillaries. The idea is that out of the blood capillaries, some blood plasma seeps out into these spaces, and here it would make the tissue fluid. Some tissue fluid gets reabsorbed back into the blood, but the remaining tissue fluid that we have here, this cannot enter into the blood capillaries again toward the venular end, but rather than it would be carried back with the help of the lymphatic vessels. Irrespective of that what the structure E, B, Z and the stuff like that is concerned, let us just look what the question is there in terms of the alimentary canal. So I have just included part C because this is relative to the alimentary canal from this diagram. They say lacteals are another important part of the lymphatic system. Like the lymphatic system has got the lymphatic vessels in it. And he is giving us the name of one of the vessel of the lymphatic system, and that is a lacteal. So they say state where in the body lacteals are found and state their function. And we know, as I have been doing it from the very beginning, that at the center of the villi, we have got this specialized lymphatic vessel whose name is lacteal. And the role of these lacteals is that whatsoever the product of the fat digestion we have got, that includes the fatty acids and the glycerol, so they would be absorbed by this and it also carries it around the body. So for 2 marks, the location is very simple. They are available within the villi of the small intestines. And the function of that would be to absorb and transport the fats or the fatty acids. Coming here, and this question appeared in the May-June of 2019. The theory paper win was 4-3, and this was question number 6B part with us. And the context of this question is relative to the enzymes that works in the alimentary canal. They tell us is that we show you figure 6.1, and this is photomicrograph of the part of the cell from the pancreas that produces enzymes that are released into the small intestines. We know it very well that we have got the pancreas that looks like this. Out of the pancreas, there emerges out a tube that is the pancreatic duct, and this pancreatic duct brings the pancreatic juice containing the pancreatic amylase and the pancreatic lipases to the small intestines. If we just look on toward its B part, and what they are telling us is that the structure labeled as transport enzymes to the cell membrane for the release into the pancreatic duct, these structures contains molecules of amylase, trypsin, and lipases. So the thing is that once the enzymes they are produced within the pancreatic cells, so these structures that are labeled as S, it would contain the amylase, the trypsin enzyme, or the lipases enzymes, and that would be released into the pancreatic duct and ultimately it would carry it to the small intestines. Let us see what the examiner wants us to do. So they gave the blanks for the total of 7 marks, where we were required to complete the sentences with the most appropriate words. The first statement is simply in terms of the general mechanism of the enzymes activity or the general nature of the enzymes, and they say is that enzymes are made of the protein and acts as dash because they increase the rate of chemical reaction but are not changed in those reactions. So we know enzymes act as a catalyst. Later they say amylase speed up the digestion of dash to dash. So we know amylase works on the starch and it converts it into maltose. If you would write glucose, that would also be correct. Then this statement from here to here is in terms of protein digestion. So they name an enzyme which is trypsin. They say trypsin continues the chemical digestion of the protein begun by the enzyme dash in the stomach. But we are aware of that trypsin is there in the small intestines. And before that, the protein digestion it happens in the stomach with the help of the pepsin enzyme. They say the optimum pH for pancreatic enzyme is greater than pH 7, and we know it very well that The pancreatic enzymes, they require an alkaline pH, so it has to be greater than 7. The bile is produced by what? We know this is produced by the liver. And then it enters into the small intestine. And over there, when it would go, so it would neutralize the stomach acid. And the stomach acid is there in the liquefied form of the food which is coming, and it is coming from the stomach. So for sure, it would have the acid in it. And when the stomach acid is neutralized, so it would provide the appropriate pH. Later, they say bile also breaks down fat by dash to increase the surface area for the action of the lipase. Now, if I want to increase the surface area of some substrate for the optimum activity of the lipase enzyme, so the bile brings about the emulsification process. So this is how you would score total of 7 marks. You just need to know the context and you just need to fill it up with the appropriate words. If we come here to question number 3, this appeared in October November of 2019 and the theory paper variant was 4.2. Here the context is in terms of hydrochloric acid, the activity of the pepsin enzyme, what assimilation is, what is the role of the microvilli, and a slight mathematical calculation. If I just begin with it, they say we have got the cells and they are there in the stomach and they excrete the gastric juice. So idea is very simple. We have got the stomach with us, and in the stomach we would have some living cells, and these cells they produce gastric juice and they would excrete them out of it. And this gastric juice, they say, contains hydrochloric acid and the pepsin enzyme. So the hydrochloric acid and pepsin, they both are relative to the protein digestion. For 2 marks, they want you to state 2 roles of the hydrochloric acid in the stomach. The thing is that whatsoever the food we are eating or the fluid we are drinking, it can have the pathogens in it. And the hydrochloric acid is one of the major chemical barrier because it kills the pathogen in the food. The other thing is that this pepsin enzymes requires the acidic pH, and the acidic pH would be provided by the hydrochloric acid and it would make the pH acidic. So the other valid point would be that this hydrochloric acid would be providing a suitable pH for the pepsin enzymes. If you forget about these two, the third valid point could be that since in the mouth the enzyme available was the amylase, so I don't want the amylase to work in my stomach. So the same hydrochloric acid have an ability to denature the salivary amylase that have come in the food from the mouth. For the 2 marks, they want us to describe the function of the pepsin. Pepsin, we know as a fact that this enzyme is there in the stomach, and the role of this would be to bring about the breakdown of the protein hydrolytically into either you would say amino acid or peptides. Now comes a very common definition for the total of 2 marks where they want us to define the term assimilation. The assimilation is that once we have digested the food and we have absorbed the food into the blood, so that absorbed food molecules with the help of the blood plasma, they would come to the cells and then they would get out of the blood plasma and it would enter into the cells. Now, once the absorbed food molecules, when they enter into the cells and then they become the part of the cells, like either it can make their cell membranes or it can make their enzymes, so that would be the assimilation. So simply assimilation is that process where there is movement of the digested food particles into the cells where they become the part of the cells. If I were to come toward the C part, they say there are stem cells in the epithelial tissue that form the lining of the stomach. The lining of the stomach simply means the cells that we would have here, okay? And they say that uh, there are some stem cells here. And they ask us that why these stem cells are necessary. What you are required to be knowing is that in my body, we have got these unspecialized cells, and these unspecialized cells, they are the stem cells. Why the stem cells are required? Because the stomach has got hydrochloric acid in it. There are a lot of muscles that are pressing on the food to be bringing about the physical digestion of the food. So over time, some of the cells of the stomach, they would degrade or they would die, and I would want to replace them if I would like the stomach to continue its proper role. This is one approach. The other approach is that I need some special cells here in the stomach, like the goblet cells, to be producing mucus and the stuff like that. So no matter if you relate the stem cells to be replacing the dead or worn out tissues, or if you relate them to be producing the specialized cells, both contexts would be right. So I would say that these stem cells, they would divide by mitosis, and as a consequence of that, they make some new cells, daughter cells, and they would become specialized cells like goblet cells that would secrete the mucus. Either this approach or the other approach would be the same, that these stem cells, they divide by mitosis to make new cells, and these cells, new cells, the new daughter cells, they would replace the cells that are worn away from the surface of the stomach or to be repairing the damaged tissue. The tissue is always repaired, and the dead cells, they would be replaced with the new cells. And thanks partly to these stem cells that we have with us. Moving toward the D part, they tell us is that the epithelial cells of the small intestine have many microvilli, and they want us to state the role of the microvilli. And the role of the microvilli is very prominent. We know they increase the surface area to the volume ratio. These microvilli, they also assist in the absorption of the digested food particles, for example, the glucose. If I were to come to the E part, this is investigation based, but the calculation was very easier for this. They happen to tell us about lactobacillus, and they say this is a type of the bacterium. Now, what the scientists did, that they carried out the investigation, and over there they wanted to see the ability of this lactobacillus bacteria, that how come it can attach with the epithelial cells that form the lining of the small intestines. If I were to make the small intestine for a while, so it looks like this. The lining of the small intestine, this has got in itself the epithelial cells in their villi, and this lactobacillus is have an ability to attach with the lining of the small intestine, and they are actually trying to see that how good it is or how much able this is to attach with the epithelial cells. How they carried out the investigation, like they took this lactobacillus bacteria and then they added it to the epithelial cells that had been grown in the petri dishes. Like we are not actually releasing this bacterium into a live human being, but rather than we would have the culture of the epithelial cells within the petri dishes, and then we would add these lactobacillus bacteria, and then we would try to see that how they were attaching with them. Further, they say that every 15 minutes, the researchers estimated the average number of the bacteria that were attached to the epithelial cells in the petri dishes. Like the bacteria which they added here into the petri dishes, every 15 minutes they were calculating that to the epithelial cells how much of the lactobacillus bacteria were attached with them. And the results they show us in this figure 3.1. I have got the time in minutes on toward the x-axis and the average number of the bacteria that got attached to the epithelial cells. Now this is something they measured, so this would be our dependent variable. Okay. I would not waste my time in making a conclusion or to be elaborating what the graph is. Before that, we just simply need to go to the question. And here they want us to calculate the percentage increase in the average number of the bacteria that are attached to the epithelial cells from 45 minutes to 75 minutes. They have given you the time. We would see that time and we would go to the graph to get the value of the average number of the bacteria, and then we would fill up that how many of the bacteria we had got at 45 minutes, how much we have got at 75 minutes, and we simply then need to calculate the percentage increase. Now reverting back to here, the first time hint that they gave us, this is at 45 minutes. So 45 is lying in between these two values. From here, I would go to the curve and then I would come here. On the y-axis, each small unit is representative of two units. So I have 72, 74, 76, and 78. So at 45 minutes, the average number of the lactobacillus bacteria attached with the epithelial cell is 78. Likewise, they gave you the other time frame, which was 75. So I would go from 75, I would come on the curve, and from here I would get my number of the bacteria, and this is 120. So I have got the average number of the bacteria at 45 minutes, which is this much, and at 75 minutes, which is this much. Now, the percentage increase becomes easier for us to calculate, and it has to be nearest to the whole number. So when you are required to be calculating the percentage increase, the final value should be bigger, and we need to subtract the initial value, which would be smaller. We would divide it by the initial value, and since this is a percentage increase, so I would multiply it with 100. And since we have to be writing it to the nearest whole number, answer we are getting is 53.8. So we would round it off to the nearest whole number, and this would become 54%. Let us come to this question, and this appeared in October November of 2019. The theory paper variant was 4.3, and for your exam, this was question number 3 with us. This is a mega question that is based on your evaluation of the digestion of the carbohydrate within your alimentary canal. Let us see what the examiner has got to tell us. They say carbohydrates are an important component of a balanced diet, and we are convinced with it that the carbohydrate, the protein, the fats, mineral, vitamins, and the other stuff they make a balanced diet. They have given us a flow chart in the figure 3.1, and they happen to show us some of the processes that happen to the carbohydrate in food that is eaten. So, if I am eating the carbohydrate in my food, so what would happen to it in the alimentary canal? So, they have presented us with the help of the flow chart. The first thing is they say that there is ingestion of the large biological molecule where we would be simply eating it up, like we would pick it up with the hands and we would try to be placing it in our mouth. Then they say that this undergoes the chemical digestion, and by the chemical digestion, what we know is that there would be the enzymes coming into the play that would break down the food chemically into the small molecules. Afterward, they say is that there happens to be the absorption of it into the blood via the alimentary canal, and there would be an increase in the glucose concentration. And then they say that this is the assimilation in the liver. Now, in simple words, these are the aspects of the nutrition where the food would be ingested, and there would be this chemical digestion with us. There would be the absorption in the blood, the increase in the blood glucose concentration, and assimilation. And these are the four major fundamental aspects of the nutrition in the human body. Let us go to the question to see what the examiner has to ask us. And as I said, this was a mega question carrying total of 8 marks, and against the 8 marks, what the examiner has to be asking us is that describe what happens to the carbohydrate in the human body between ingestion when you have eaten it and assimilation, when it would become the part of the liver. And they want us to use information in figure 3.1 as a guide. Now, let us just try to crack some of the points with context of ingestion of the large biological molecules. So we know that the ingestion would be happening in the mouth. And when we talk about the mouth, so in the mouth, what we actually know is that there would be the happening of the mechanical and the chemical digestion of the carbohydrate. Okay. Now, why the mechanical digestion? Because within the mouth we have got these incisors, canine premolars, and the molar teeth. So what they happen to be doing is that this carbohydrate that we have eaten it up, it would be broken down into the small pieces. Same within the mouth, what we know is that there would be happening of the chemical digestion where the salivary amylase enzyme would come from the salivary gland and it would break down the carbohydrate after starch into the maltose. Now, if we happen to be talking about the chemical digestion now, the first chemical digestion, this would be happening here in the mouth, and the second chemical digestion for the carbohydrate, this cannot happen in the stomach. Like while the food is being broken down or the carbohydrate is being broken down physically and chemically in the mouth, so it would be converted into a spherical kind of a shape, and that is the bolus with us. And then it would pass through the esophagus and the stomach. And in the stomach, what we would know is that there would be no happening of the chemical digestion, but the stomach wall has got the muscular walls, so that can break down the carbohydrate physically into the small pieces. Finally, the chemical digestion of these carbohydrate would be completed in the small intestines. Why the small intestines? Because from the mouth, what's coming is the maltose, and the other thing coming up is the leftover starch. So once this would be there in your small intestine, so the pancreas would be sending pancreatic amylase enzyme, and that pancreatic amylase would break down the leftover starch to the maltose. Finally, we have got the maltose with us, and in the villi of the small intestine, we have got the maltase enzyme, and that would break down the maltose to the glucose. Now, you see, they say is that there is an increase in the blood glucose concentration because after we have done the complete chemical digestion, so now we would absorb them via the villi into the blood capillaries with the help of the diffusion and the active transport. And the carbohydrate being absorbed is now not a complex carbohydrate, but it would be the glucose with us. And for sure, when the glucose would be going from the villi into the blood capillaries and it would be carried to the liver with the help of the hepatic portal vein. So the blood glucose level would increase. And then the pancreas would be stimulated to be producing the insulin hormone. And the insulin hormone would now come to the liver where excess glucose would be converted into the glycogen, whereas the rest of the glucose would be used up for the process of the respiration within the liver. So this is how I have told you each and everything with respect to the points which they have given us in the flow chart. So let me take you to my answer, that how your answer should appear if this appear to be in your upcoming exams. So we would say that initially this carbohydrate it would be ingested in the mouth, and in the mouth it is broken down into the small particles, and that would be the mechanical digestion by the teeth. At the same time, the salivary gland would come into the play. They would release saliva out of it, and that saliva we know has an enzyme salivary amylase in it. And that salivary amylase would break down the starch that we have in our food and it would be converted into the maltose. While the food is there in the mouth, so it is converted into a spherical shape structure which is bolus, and then it would go to the stomach with the help of the esophagus. In the stomach, as I have mentioned above, that there is no chemical digestion of the carbohydrate, but rather than in the stomach, we have got the muscular walls and that churn and break down the carbohydrates physically. Finally, this carbohydrate would be coming to the small intestines, and from the mouth we have got the leftover starch, and this would be broken down to the maltose, but not by the salivary amylase, but rather than this would be done in the small intestines with the help of the pancreatic amylase. Now, you see, we have broken down the carbohydrate, all of it to the maltose, but the maltose further would be broken down by the maltase enzyme, and that enzyme is available within the epithelial cells of the villi. And once the maltose is acted upon by the maltase enzyme, so we would now have the glucose. So here the chemical digestion of the carbohydrate has ended. Now we would take the glucose into the blood capillaries of the villi, and for that purpose, the diffusion and the active transport would be contributing their role. After we have taken them into the blood capillaries of the villi, so now the glucose would be carried to the liver, and that too with the help of the hepatic portal vein. Once the liver has received the glucose, so the glucose level is high, so from the pancreas, the insulin would come and that would convert the glucose to the glycogen. And what we know is that some of the glucose still would be required to be used in the respiration, that the liver cells would be aerobically respiring and would release the energy out of it. So this is how you would score a total of 8 marks for this. Let us go to its B part, and the marks were 4. What they happen to be telling us is that mineral salts are another important component of a balanced diet. They want us to state the importance of the calcium ions and iron ions in the balanced diet. So that means in each heading we should be telling him two importance or two critical point in terms of their importance in our balanced diet. So these are the facts that the calcium ions, they help to strengthen the bones and the teeth. And also, if you would like to write some other alternative points, so that could also be that it is important for the conduction of the electrical impulses. If you don't like this point, then as an alternative valid point, you could also be writing that this calcium ions are also important for the muscular contractions. If we talk about the iron ions, so we know it very well that they are available within the red blood cells, and they happen to be binding with the oxygen gas and would transport it. The other point would be that they help prevent the anemia where the red blood cells become deficient in the body. As an other valid point, you could say that this iron ion, this is found within the red blood cells because the presence of the iron ion within the red blood cells is also the importance of it. Moving onward, here let us come to be discussing this question from May-June of 2020. The theory paper variant was 4-1, and for your exam, this was question number 3, B and C part. Let us see what the examiner has to tell us. They say that we present you with figure 3.2, and this is a drawing of a cell from the lining of the small intestine. Now, what we know is that the lining of the small intestine is basically the villi with us. Further, they tell us is that the lumen is the space inside the intestine where the food is digested. Now, as I have been telling you from the very start that here, if we have got this small intestine, then this empty space within the small intestine is the lumen where the food is getting digested. And if I talk about the lining of the small intestine, so these are the small finger-like projections that we call them as the villi. And from the villi, they are presenting you a cell out of it. And this is very evident, like since I see a lot of these micro projections, so these are the microvilli with me. If I talk about C organelle that is presented here, so this is the mitochondria. If we happen to be looking on toward the B structure, if I were to zoom it more, so I have got an extended network of the membranes having ribosomes on it. So this would be rough endoplasmic reticulum. So overall, this is an epithelial cell of the villi of your small intestines. For the total of 6 marks, the examiner is interested that you state the name of the three labeled structures that are given in figure 3.2, and we are required to describe the role of each structure in the intestinal cell. So, as I have identified them before, so A was microvilli, and this increases the surface area to the volume ratio for the absorption process. The mitochondria, they would release the energy with the help of the respiration process, and that would be moving the food molecules from the lumen to the inside with the help of the active transport. If we were to talk about the B, which is rough endoplasmic reticulum, so its role would be to bring about the synthesis of the proteins. And with context to the epithelial cells, those proteins would be the enzyme that later become incorporated or they embed within their cell membranes and they would bring about the digestion of the double sugars, which are the maltose, lactose, and the sucrose. So if I were to simply read my answer for you, so as I've said, is microvilli, this increases surface area to volume ratio for absorption by word diffusion. Or you could also say that these microvilli they happen to have some transport proteins in them that facilitate the movement of the molecules for active transport. Now, against microvilli, either you write this point with it or you write this point with it or the point relative to the presence of enzymes within the cell membrane. The B was rough endoplasmic reticulum, and this is the site of the protein synthesis. That's it. C we have got is the mitochondria, and this releases energy by aerobic respiration for later to bring in the food molecules with the help of the active transport. If I were to go toward its C part, and here they talk about the bacterium, which is Vibrio cholerae, and they say that this cholera bacteria, once it's come in the small intestine, this can survive not only in the small but also the large intestine. The bad thing about this bacteria is that this releases the toxin, and once the toxins they get released out of the bacteria, which is Vibrio cholerae, so it can interact with the receptors on the surface of the cells. And you see the cell is the same, which is the epithelial cell of the villi. Now, in the figure 3.3, they are showing us the effect of the toxin. And the arrow which they gave us here, and at these points and these points, they actually indicate the direction of the movement. So if I were to simply like be telling you the mechanism of it, and for sure by that you would get the answer. Here the bacteria is coming, okay, and the bacteria is releasing the toxins out of it, and the toxins are presented with it. The toxins they are getting into the epithelial cells of the villi, and once the toxins they are getting in, so what they happen to be doing is that they would pump the ion X, and that ion X is the chloride ion. And when the chloride ions, their concentration happen to be increasing in the lumen of the small intestine, so this automatically would be decreasing the water potential here. And once the water potential decreases here in the lumen, so the water level or the water potential in these epithelial cell, this increases. And as a result of that, the water would move out of these epithelial cells. It would come into the lumen of the small intestine, and by that, the person would develop the diarrhea. Okay. So this is the overall explanation of the question which the examiner has asked us below. For the first 1 mark, they tell us is that the toxin stimulates the secretion of X out of intestinal cell. This X ion, they want us to tell its name, so this is the chloride ion. For another 4 mark, they are asking us that describe the effect on the body of the secretion of ion X into the lumen of the small intestine. Like if this chloride ion is going out, how can this impact us? This is what we are required to be describing it. And I have given you a clear idea of that what needs to be written here. So we would simply say that when the chloride ions would come into the lumen of the small intestine, so it would simply lower the water potential there. And this would cause the water to move out of the cells by osmosis, and this would lead to the development of the diarrhea. The person, as a result, would suffer from dehydration, and the essential ions like the chloride ions and the calcium ions and the other important ions they would be lost in diarrhea. So this is how this toxin which was released from the cholera bacteria, this can have a very harmful or the negative effect on our alimentary canal. Moving onward to this part, and this came in the May June of 2020. The theory paper variant was 4-3. And for your exam, this was question number 1, A, B, and C part. For the first, they say that state 3 uses of energy in the human body. And we are very much aware of this fact that the energy would be used for the growth where we increase in size. It would be for the movement aspect, for the nerve impulse conduction. If you don't want to write these points, the other point could be that it would be for the reproduction, for the excretion scenario. And now to the B part, here your identification of the components of the elementary canal would be required. And this is simply the part of the digestive system of the human which they show us. Like if I were to zoom in to be identifying these structures for you, so A that we have here, this is esophagus. The B that we have got is the stomach. And the K that we have here, this is the liver. The L that we have got here, so this is the gallbladder. The C structure that is shown here, so they are the pancreas. And if I were to just come here, this part H, so this is the first part of the small intestine. That would be the duodenum with us. And the D that we have got, so this is lying somewhere here. So this is toward the end of the small intestines. But we would either consider it as ileum. And then we have got this F now, this is the appendix with us, and this G that we have got, so this is the large intestine, and the E that is shown here, so this is the rectum where we know that the undigested food particles in the form of the feces would be stored here for some short period of time. Now, let us see what the examiner has to be asking us. And you see 6 marks, and they want us to be completing the table 1.1. And for our benefit, they have completed the first row. Like the function is to push food into the stomach. So this is the esophagus, and the latter from the figure is 1.1. Now they say is that what brings about the assimilation of the amino acid to produce the plasma protein. So the assimilation, we are aware of this fact that this would be happening within the liver, and the latter would be K with us. Moving onward, they ask you to be telling him the storage of the bile. And what we know is that the bile is excreted out of the liver, and it would be temporarily stored within the gallbladder. And the letter that I have with me, so this is L with us. They say that what brings about the secretion of the insulin, so this is fact that they are the pancreas. And if I just take you to be identifying the letter, so this is letter C with us. Likewise, they want you to tell them that what brings about the absorption of the fatty acid and the glycerol. So the fatty acid and the glycerol, they gets absorbed from the small intestine. So here you have the choices, like either you write it as H or either you write it as D. You would score full marks. Next, they asked you to be telling them that what brings about the secretion of the pepsin. So this is a factor, this is the stomach. And the latter that we have got for the stomach, so this is B with us. The last is that where do the digestion of the starch happens. Now we are very much aware of this fact that the digestion of the starch would happen in the mouth or within the small intestines. So again, this is your choice. Either you would say H or either you would say D. And in both cases, the answer that you are required to be writing here would be the small intestines. If I were to move toward the C part, they ask us that describe the role of the liver in the recovery from the oxygen debt after the strenuous exercise. So once we have done a strenuous exercise in our body, there would be the production of the lactic acid. And once we rest, so in that resting phase, this lactic acid would be getting out of the muscles into the blood, and the liver would absorb it. Once the lactic acid has arrived into the liver, so it would break it down into the carbon dioxide and the water with the aerobic respiration process, or can convert it into the glycogen as well. Moving onward, here we have got this question on the teeth structure from October November of 2020, and the theory paper variant was 4-2, and this was question number 2 with us. They show us figure 2.1, which is a vertical section of a human molar teeth, and its surrounding structures. Now, if you have to be focusing here, they say is the enamel with us, which is the white outer protective layer of the teeth. In the B that we have here, so this is dentine. The C that shows some either nerve endings or the blood capillaries here. So this would be the pulp cavity. And the D that we have here at this point, so this is the gum with us. Simple as that. So A was enamel, the B is dentine, the C that we have is the pulp cavity, and the D that we have here, so this is the gum with us. Moving onward to its B part, the total marks for this part were 3, and the examiner wants you to describe and explain the function of the molar teeth. No matter what type of the teeth come here, no matter if this is molar, canine, or premolars or incisors. The idea is very simple that these teeth in the mouth they would bring about either you would say physical or mechanical digestion. And you know it very well that these are the molar teeth and they are located at the end of our jaws. So their role is to crush the food. And when they would crush the food, they would split it up into the small particles. And as a result of that, the surface area of the food particles due to the crushing by the molar teeth, this would be increased, and now it would allow the salivary amylase enzyme that would be coming from the salivary gland to break it more effectively with the help of the chemical digestion. So this is how you would score total of 3 marks out of it. Now coming here to this past paper question. From October November of 2020, the theory paper variant was 4-3, and this was question number 2. And the theme of this question is to assess your idea of that how good you are with understanding the aspects of nutrition like the ingestion and the digestion and the absorption and the assimilation. And the next part of this question was related to what happens within the stomach. So, for the A part, they say a person ate a meal and it has starch and the fat in it. And in the figure 2.1, they show us some events that would occur after ingesting the meal. Now, what they want us to be doing is that we need to put the events in figure 2.1 in the correct sequence, and they have done the first one for us, and that is R. And if we were to look on R, so that is the breakdown of the large food particles by the teeth. Now, what we know is from this that if there is happening of the physical digestion, then we would look for the other point in terms of the chemical digestion in the mouth, and it's written as T that there is secretion of the amylase from the salivary gland that would be acting upon on this. So immediately after R, we would be writing the T here. In the mouth, the food would become into the spherical ball-like structure that is a bolus, and now we would move it to the other part of the alimentary canal. And you see, the next point that we have got here is S, like the movement of the small food particles through the esophagus. So this would come after the T. So the S would come here. Now, once we have moved the food through the esophagus, there would be happening of the absorption of the nutrients in the villi. So the P would come, and the last point would be that there would be assimilation of the fatty acid, like they would become the part of the liver cell. So it would be Q that would be coming at the end. If I were to take you to the B part, what they ask is that the stomach lining it contains the cells that secretes the proteins. For 2 marks, they want us to state the name of two cell structures that are involved in making or secreting the proteins. Now, in the making of the protein, what we require is a nucleus because the nucleus has got the genetic material stored in it, and that genetic information or the material is what is required for the making up of the protein. The other important thing that we require for the making up of the protein would be the rough endoplasmic reticulum, or we say would be the ribosomes. As an alternative valid point, we can never forget about the mitochondria because this entire protein synthesis process requires energy, and that would be provided by your mitochondria. So I have written nucleus and the ribosomes, If you forget about any one, you can write the mitochondria as well. If I were to take you to this second part for the total of 3 marks, they say state the names of one component of the gastric juice and describe its function. From the IGCSE's level, what we know is that the gastric juice would have the hydrochloric acid, or it would have the pepsin enzyme in it, or the pepsinogen enzyme in it, but it's more simple to write it in terms of the hydrochloric acid. And we know that this hydrochloric acid, this would kill the pathogens or the disease-causing organisms that we have mistakenly eaten in the food. Other than that, the pepsin enzyme that we have in the stomach, this requires the acidic environment. So that acidic environment again would be provided by the hydrochloric acid. In the mouth, we had the food and the salivary amylase was added into it. And now that has to get denatured in the stomach for the pepsin to be doing its work properly. So the same hydrochloric acid, this would denature the salivary amylase enzyme. Moving onward, we have got this third point, and they tell us is that there are the goblet cells in many parts of the alimentary canal. For 1 mark, they want us to state the substance that the goblet cells secrete, and this is a fact that the goblet cells releases a sticky substance out of them, and that is mucus with us. For the C, they tell us is that emulsification of the fat occurs in the alimentary canal. Describe the process of emulsification of the fat. So in the emulsification, the large fat droplets or the fat molecules that we have eaten in the diet, when they are acted upon by the bile coming from the gallbladder, so when they break down into the small fat droplets or to the small fat globules, that decreases their surface tension, increases their surface area, and makes it easier for the lipases enzymes to be breaking them down into the fatty acid and glycerol effectively. So this would be the emulsification with us. The next thing they say is that state the name of the organ in the alimentary canal where the fats are emulsified. So this again is the fact that this happens. This actually is initiated as well as completed within the small intestines. For the last 1 mark, they want you to state the name of the structure in the villus where the digested fats are absorbed. So if we are absorbing the products of the fat digestion, so at the center of the villi, what we have is a specialized lymphatic vessel, and that is the lacteal with us. Now, coming onward here to October, November 2021, the theory paper variant was 4.2, and this was question number 1 for your exam. They tell us is that figure 1.1, it shows us several villi from the ileum, which is a part of the small intestine. The small intestine has duodenum, jejunum, and ileum, and here they are talking about the ileum. And for 1 mark, they are asking us that state the name of one other part of the small intestines, like ileum they have given. So apart from that, we have the duodenum and we have the jejunum. So whatsoever comes in your mind, you may write it down. Now, if I were to be coming toward this 1.1 diagram, so what they show us is the lining of the small intestine. And the lining of the small intestines, they are the villi with us. And if you were to see, like within the villi, they are showing you the cells here, this cell and this cell and this cell. So they are the goblet cells and they secrete the mucus out of them. And if we were to come here at the center of the villi, so they show us a lymphatic vessel, that is the lacteal. And they say is that this part that is marked here, so they would show us an enlarged section of this. Okay, so this part right here, this part. So its enlarged diagram is shown here. And if I were to zoom in, so they show you the epithelial cells here. And if you were to look clearly, these epithelial cells, they have these tiny projections on the top of them, and they are the microvilli. And at this point, what they are Labeling for us is that this is a goblet cell, and this goblet cell releases the mucus out of it. If we were to come on toward the questions for the B part, they say that the epithelial cell of the villi, it absorbs the nutrients by the diffusion and the active transport. When the food would be coming in the small intestine, so in the lumen, the carbohydrate and the protein and the fats, they would be completely digested. And now they would need to be absorbed into the body with the help of the diffusion and the active transport. For 3 marks, they happen to be asking is that describe how the active transport differs from the diffusion. So what we know is that the active transport would move the particles against the concentration gradient. The diffusion happens to move them down the concentration gradient. The active transport would move the particles by using the energy which is released from the mitochondria. It never happens in the diffusion. And at the same time, whenever something is being moved against the concentration gradient with the help of the active transport, so there would be the involvement of the transport proteins. So since the question was in context of the active transport and how this is different from diffusion, so I have written 3 points for description in context of the active transport only. Moving onward for 2 marks, they say is that explain the importance of the microvilli, which are shown in figure 1.2. And our idea is very clear that if I have the epithelial cells, and on the top of the epithelial cells, if we have these microvilli on them, so they would increase the surface area to the volume ratio. When the surface area to the volume ratio is increased, so this would result in a faster absorption by the diffusion process. Why I have written 2 points? Because the marks were two. Moving onward to the C part, and they have shown us the diagram of the goblet cell here. You've seen like there was a goblet cell present here, and now here the question comes that the goblet cells they provide protection for the epithelial cells that line the small intestines. For 1 mark, they want you to state the name of the protective substance which is produced by the goblet cell, and we know that sticky or protective substance is the mucus. The second part of C is little technical, and they say is that why a protective substance is necessary in the small intestines. Now the idea is very simple because in the small intestines there happens to be the presence of the digestive enzymes, right? And at the same time, there is a possibility that the food that has come into the small intestine, it can have some disease causing microorganisms in it or the pathogens in it. Moreover, you see that the small intestine they are always in a state of peristalsis where it muscles that we would have in the intestine compared to the stomach, the musculature or the muscles within the wall of the small intestines are less, but still they keep on pushing on the food. So you see that when the food would come in contact with the small intestine, so this can damage the inner cells of the small intestine. So this is something we don't want. Now, in order to be cracking 2 marks, but we would tell him that when there would be the presence of this protective substance mucus, so this would save our small intestines from the physical damage, and the physical damage can be due to the movement of the food. Other than that, within the lumen of the small intestine, there happens to be the presence of the enzymes, and I don't want that these enzymes start digesting the cells of the small intestine. So the mucus would save the self-digestion of the cells that are lying here. And last, what we could say be saying is that there happens to be the presence of some pathogens within the food. So when the mucus would be available, those pathogens would have a less chance to be attaching with the cells of the small intestines or to be damaging it. Now, for the D, what they say is that figure 1.1, it shows a lacteal at the center of the each villus, and they want us to describe the role of the lacteals. If I go back to figure 1.1, so at the center we have this lacteal, and the role of the lacteal is very simple. We know it very well that this lacteal is a specialized lymphatic vessel. It has a lymph in it, and the role would be to absorb the breakdown product of the fats, that is a fatty acid and is a glycerol. And the other role would be that not only they are absorbing it up, but they would carry it around the body with the help of the lymphatic vessels. Our blood capillaries are not in the position to be absorbing these fatty acids. So when they go into the lymphatic vessels, which is the lacteal, later they drain them back into the blood. We were to come to the E part, they have given us multiple structures here, right? Like the gallbladder, endoplasmic reticulum, intestinal epithelium, and the ileum, and we need to fill them up on the basis of their level of organization. Now, what we know is that this gallbladder, this is the organ with us. The endoplasmic reticulum that basically has the ribosomes attached on them, since this is present within a cell, so it would be a cell structure. If we talk about the intestinal epithelium, so we would not regard it as a cell but rather than a tissue. If there was something written as epithelial cell, then we would have considered it a cell, but intestinal epithelium would be regarded as a tissue. If we talk about the ileum, so although it's a subpart of the small intestine, but we would be considering this as an organ. If I were to move here, so this is coming from the May-June 2021. The theory paper variant was 4.3. And for your exam, this was question number 1. And here the examiner was interested to evaluate at how good you are with the composition of the biological molecules that you eat. And if they are broken down, so what products you would be receiving and what enzymes would be involved. So overall, this is enzyme related question, but the enzyme that contribute their role in the digestive process in the alimentary canal. They say is that the main component of the milk is a liquid that is an important solvent in our body. Although makes a major part of the milk, but is acting as a solvent in our body, and we know this cannot be other than the water. For the table which they have given us, what we see is on my left hand column is the nutrient, then we need to be writing their elements, the enzymes, and what would be the product of the digestion. So we know that the protein, the elements would be carbon, hydrogen, oxygen, and nitrogen. If the enzyme has to be breaking down the protein, so it would be a protease enzyme. And when the protease would work on the protein, so we would get the amino acids out of it. For the fats, the element is only carbon, hydrogen, and oxygen. The enzyme breaking down the fat would be the lipase enzyme, and the products of the digestion would be the fatty acid and glycerol. For the last, lactose sugar, they just did not fill this one column right here. So we would say that if the lactose has to be broken down, then the enzyme acting on it, it would be the lactase enzyme with us. The lactase is a disaccharide enzyme, and that would be available within the cells of the epithelium. Likewise, if the word would be maltose, enzyme would become maltase. If the word here would be sucrose, enzyme would become sucrase. If I were to come here to the B part of it, so they ask you is that milk also contain vitamins and minerals, and they are asking for the importance of vitamin D to be included in the nutrition of the children. Vitamin D is important with context too, that if I am taking calcium, but if I am not taking vitamin D along with it, so the absorption of the calcium would be compromised. So vitamin D brings about the absorption of the calcium into the bones. At the same time, this vitamin D is important for strengthening or making the teeth and the bones strong. If there is deficiency of the vitamin D, then the infants or the children they suffer from the rickets where their bones become soft and they tend to bend under the weight of the body. So if vitamin D is available in the diets, it would prevent these children from getting rickets. And then for another 2 mark, the examiner wants you to tell him the importance of iron in the diet. And iron is a part of the hemoglobin protein. And this is necessary. Why? Because the hemoglobin protein, if it would not have the iron with it, so it would not be able to bind the oxygen gas with it. Okay, so simply we would be saying that This is important for the formation of hemoglobin. And once it binds with the hemoglobin, then it would be transporting the oxygen gas around the body. As an alternative, other point, you can also keep this in your mind that if iron is available in the diet, so you would not suffer from anemia. Moving onward, here we come to October November 2021. The theory paper variant was 4-1. And for your exam, this was question number 3B with us. What they have to be telling us is that the scientists they use enzymes in the bile in the early development of biological washing powder. Now, how do we develop the biological washing powder? This would be coming in the biotechnology, as we have been talking about the role of the biles in the body, so they are the same. So I will just simply read it out for you. We have done it in so many questions above. It brings about the emulsification of the fat, and then it provides the alkaline environment for the intestinal enzymes. The food that is coming from the stomach into the small intestine would have the hydrochloric acid in it, so it would neutralize the hydrochloric acid. And when the bile would break down the large fat molecules into the small fat droplets or the fat globules, so this would increase their surface area so that later the lipases enzymes can work effectively on the fat globules. Coming here to May June of 2022, the theory paper variant was 4-1, and for your exam, this was question number 3, A, B, C, and D part, and this is related to your understanding of the structure of the alimentary canal. What they tell us is that the pancreas is an organ. It has a role in digestive and the hormonal system of the humans. The digestive roles of the pancreas are that they would be producing the pancreatic amylase, it would be producing the trypsin enzyme for the breakdown of the proteins. This would also be producing the lipases enzymes for the breakdown of the lipids. In terms of the hormonal control, the same pancreas would release the insulin and the glucagon hormone. They tell us is that we show you this figure 3.1, and this shows us a part of the alimentary canal and some of the organs that are associated with it. So if I were to just simply highlight them up, so this part here, this is the diaphragm, this is esophagus, this that we have here is the stomach. This J that we have got is the liver, and this K which is attached to the liver, so this is the gallbladder. And if we talk about this part, this is the pancreas. And when I get out of the stomach, so this first part of the small intestine, that is the lateral, so this is the duodenum with us. Or you can simply write it as the small intestine. And for 3 marks, they want us to name the organ J, K, and L. So J is the liver, the K that we have here is the gallbladder, and the L that we have got. So it's your choice. Either you write it as small intestine or you can be writing it as the duodenum. If I were to move toward the B part, so this is the same thing that they have presented above, that the pancreas is releasing the hormones and the enzymes, and that would come into the pancreatic duct. Okay, like if I have got this pancreas, they would release the enzymes, so they would come into the pancreatic duct. The hormone they would actually go into the blood plasma. So that is why they want us to fill up the table which they have given below, where we need to be stating the hormones and the enzymes that are secreted by the pancreas. And as I have said above, that the hormones secreted by the pancreas are two major hormones. One among them is insulin. The other one among it is the glucagon. If we talk about the enzymes that would be secreted by the pancreas, so you write it as amylases or carbohydrates. You can write it as proteases. Or if you become specific, you would say trypsin. And then the last enzyme that is released out of the pancreas into the pancreatic duct. So this would be the lipase with us. The next part that we have is same in terms of the movement of the chloride ions. Now, what they have to be telling us is that the chloride ions they move along the pancreatic duct. Like if I have these pancreas with me, and then there comes the pancreatic duct. So the chloride ions they move along the pancreatic duct. They tell us is that there are proteins which are CFTR proteins and they are available in the cells that line the pancreatic duct and they move the chloride ions out of the cells into the duct. Now, here happens to be the presence of some cells. And these cells, they have got these CFTR proteins in them. And the role of the CFTR proteins is that they would excrete the chloride ions out of them so that they come here into the pancreatic duct. And they have shown us the diagram of the same cell that are lining the pancreatic duct, and we see it here, and they have given a key where the chloride ions are being pumped out, and they are coming here into the pancreatic duct, and this is the cell which is the pancreatic cell with us. Now, for 3 marks, what they have to be asking us is that explain how the CFTR protein moves chloride ions across the membrane of the cell shown in figure 3.2. Now, what you have to be assuming at this point is that these chloride ions, now since they are continuously being pumped out, and considering they have a charge on them, so this would be done with the help of the active transport. And for moving them, there has to be the release of the energy from the mitochondria, and wherever the ion is being moved, so they would also require some transport protein that would facilitate the movement of these ions as they cannot cross the cell membrane directly, but rather than they require the presence of some transport proteins. So for 3 marks, how the answer should be written as that they move the chloride ions against the concentration gradient. And how? By using energy which would be released by the mitochondria with the respiration process. And when these chloride ions they would move, so ultimately these proteins that we have here, which are the CFTR protein, so these proteins they would change their shape. And when they would slightly change the shape, so it would help or it would facilitate the movement of the ions out of the cells and finally coming into the lumen of the pancreatic duct. Coming toward the last part, what they say is that once you have moved the chloride ions into the pancreatic duct, so this also causes the water to move from the cells into the duct, and that would help to flow the liquid in the duct. Now, the idea here is that again, this is the pancreas, this is the pancreatic duct, and once we have pumped the chloride ions out, so this would also cause the water to move out. And when the water is moving out, so any of the pancreatic juice that we have got, so this water would allow it to move toward the small intestines. For 3 marks, they say is explain how the water moves from the cells shown in figure 3.2 into the pancreatic duct. Like they are asking us that how the water moves here out of the cells into the pancreatic duct. So the concept is that previously they were pumping the chloride ions out. When the chloride ions would be pumped here, so their concentration would increase, and automatically in the pancreatic duct they would lower the water potential. As a consequence, the water potential in the pancreatic cells would increase. So here we would have now higher water potential. On the outside of the cells in the pancreatic duct, we would have lower water potential. And as a result of that, the water would move from its higher water potential in the cells to the lower water potential that we have in the pancreatic duct, and it would be done down the water potential gradient with the help of osmosis. So if I were to read the answer, so we would simply say that the presence of the chloride ions, it would lower the water potential in the lumen of the pancreatic duct. And that causes the water to move out of the pancreatic cells by osmosis and it would be coming down the water potential gradient. And in doing so, it would also cross the partially permeable membranes. Coming toward May-June of 2022, the theory paper variant was 4.3 and this was question number 4. And here your idea of the identification of the elementary canal was tested. As it themselves says that this figure 4.1 we show you the diagram of the elementary canal. So if I were to be making you identify these parts, so here we have the mouth, the number 2 labeling is the stomach, and then the third that letter that we have here, the third number that we have here, this is small intestine. Now, since this is the first part of the small intestine, we can be calling this as the duodenum of the small intestine. And the fourth that we have here, so this would be the ileum, which is the last part of the small intestine. The five that we have here, this is the large intestine, and the six that we have got, so this is the anus with us. Now, what the examiner wants us to be doing is that we are required to be completing the table where we need to name the organ that are listed above and what processes would be happening within these organs, and then we have to use the key. The A would be for absorption, C for chemical digestion, E for egestion, I for ingestion, and M for the mechanical digestion. And as we have identified a while ago that the letter 1 that we have with us, so this is the mouth, and the mouth, we all are aware of this fact that they are responsible only for the ingestion, right? Because they show you the very start of the mouth, not here. If this was here, so we can be writing it in terms of the mechanical and the chemical digestion. Now, this is just the beginning of the mouth, so we would just relate it to the ingestion. If we talk about the second letter, so it was the stomach. And the stomach has got the role in the mechanical digestion. Why? Due to the presence of the muscles. The chemical digestion with respect to a scenario that there is presence of the pepsin enzyme. And some chemicals or molecules, they have the tendency to be getting absorbed through the stomach as well, so we can use the letter A as well. As far as the first part of the small intestine, which was the third number, so it was the duodenum with us. And we know that this brings about the absorption and also slight mechanical digestion as well due to the presence of the muscles that would be pushing on toward the food. And we can never forget about the C letter because the duodenum is the site of the chemical digestion as well. The fourth one that I identified above this one, so this would be the ileum with us. That is the third part of the small intestine. And this brings about the absorption and the chemical digestion, and not the mechanical one because the musculature or the muscular content in the ileum is less. The fifth part that we have, so if you become specific, like overall from here to here, this is large intestine. But if we specifically talk about this, so this is colon with us. And the role of the colon is absorption of the water. And if we talk about the sixth letter, the sixth number that we have, this which was here, so this only has got a role in the removal of the undigested food particles that happen to exist there in the form of the feces. So it would be E only, and that would stand for the word egestion. Likewise, this other part of this question, this is related to the structure and the functions of the villi. And 4.2 that we are given here, so this is a very good diagram of the lining of the small intestine. And now this is a single structure that is being shown here. So we would call this as the villus. What they tell us is that the arrow that we show, this indicates the direction of the blood flow. Okay, like the arrow is like in what direction the blood is flowing. If I were to quickly highlight the parts that we have within the villi, so if you see that this P is located at the center, now this would be the lacteal with us. The Q that we have got, so they are the epithelial cells, and these epithelial cells, they have the microvilli in them. They also have the transport proteins in them. They have the abundant mitochondria in them. All of them are related to the absorption and increasing the surface areas and the stuff like that. If I were to talk about our, so this is a blood capillary in which the blood flows continuously and ultimately would help us in the absorption of the glucose and the amino acids. Now, if I were to talk about the use, so for sure, if we have the blood capillaries here, so this would be an artery or an arteriole. The S would be venule, and this is a lymphatic vessel that would be taking the dissolved or the absorbed fatty acids away from the villus. Now, if I were to just take you for the total of 6 marks, the examiner said that you are required to describe the structure of the villus and you have to be relating it to its role in the alimentary canal. We are advised to use a letter given in figure 4.2 that would help us to support our answer. Now, since the marks are 6, so we need to be writing just 3 description and then a slight their association with their role. So the P that I identified above, so it was lacteal, and it would absorb the fatty acids and glycerol. The T that we have got here, like that, would be draining the lymph away, having the fatty acid and glycerols in it. So this is the lymphatic vessel. So we would say that it would return the lymph containing the fatty acid and the glycerol back into the blood. The cue that we were given here at this point, so these were the epithelial cells, and as I said, that since they have the microvilli in them, so they would increase the surface area to the volume ratio. Now, at this point, you would score total of 6 marks. If you want to write some other points with context to the letters, so we can talk in context of R. And as we have identified above, the R was a blood capillary. We would say that it absorbs glucose and amino acid. Or other than that, you could also say that they are one cell thick and that would decrease the distance over which the diffusion has to occur. The third point that we can write in terms of the blood capillary would be that in them the blood flows continuously. And what you have to be assuming is that if the blood is flowing through it continuously, then it would help to maintain the concentration gradient difference that would lie in between the food particles that are here in the blood capillaries and that are available outside of the villi or the villus that would be there in the villus in the lumen of the small intestines. Likewise, the Q that I have given here as an alternative valid point, this is the epithelial cells, and I have been saying from the very beginning when we started doing the topicals that these villi also have in their cell membranes the disaccharidase enzymes like the maltase, lactase, and the sucrase enzyme. Now, since I have written the maltase enzyme here in context of the epithelial cell, which is Q, so I would simply say that it would break down the maltose into the glucose. So this is how you would have scored total of 6 marks for this. If you come here to October, November 2022, the theory paper variant was 4-1. And in your exam, this was question number 1. And here the approach to be testing was your ID in terms of the structure and the function of the teeth. What they say is that we show you figure 1.1, and this is a side view of the human skull. And they have shown you 4 type of the teeth in the jaw. Okay, so we know at the very front we have incisors, then the pointed teeth are the canines, then we have these premolars, and then we have got the molars. Then they show you the upper jaw, and then they show you the lower jaw. For 1 mark, they want you to state them the function of the human teeth. So simply like they bring about the mechanical or the physical digestion of the food. Now for 1 mark, they want you to state the name of the visible outer layer of the teeth. And this is something we see daily in the mirror that the visible outer layer of the teeth, this is enamel with us. Now coming on to word October, November 2022, the theory paper variant was 4-2, and this was question number 2 with us. Again, your approach in terms of the identification of the parts of the alimentary canal and how come the enzymes within them are working. So it was tested. There would be a question that is graphically based, so that they related them to the enzymatic activity. So they say is that figure 2.1 is a diagram of the human alimentary canal and associated organs. The associated organs are not directly the part of the alimentary canal, but they contribute their role in the digestion. So we discuss them along with it, like the salivary glands, the liver, and the gallbladders. So all of them, they are the accessory organs. Also the pancreas. If I were to quickly highlight the identification of the numbers given here, so the one that is coming here, so they are the salivary glands. The two that we have here, so this is the esophagus. The third number that we have here, so this is the stomach. The seventh number that we have got over here, so this is the liver. The fourth number are the pancreas. The fifth, you generally call this as the small intestine, but this would be the second part of the small intestine. Like this is duodenum, so it would be jejunum with us. And if we talk about the sixth number that is coming over here, so this is large intestine generally, but if we become specific, so this is colon with us. What the examiner has to be asking is that we need to be completing the table where we are required to be cracking the name of the enzyme, the organ where the enzyme would be secreted, the number they have already given us in the table, like 134, we just need to fill the last number, the substrate the enzyme would be working on, and the product or the products of the digestion. Now, the first number that they gave us was number one, and they say that the enzymes being released out of one, that is amylase. So the organ that produces the enzyme. So if you see this letter one is producing amylase. So these are the salivary glands, and we know that these salivary glands, once they produce amylase, so the substrate they work on is the starch, and we would get maltose out of it. For the second, the number they gave us, and this was 3, and they say that the substrate on which the enzyme is working is the protein. So I would simply say that if the protein they would be broken down, so for sure we would be getting amino acids out of it. The third letter that we have with us, so this was the stomach with us. And at the same time, the enzyme that would be produced in the stomach, so this is a fact, it is pepsin. For the third, they wrote here that this is lipase, and we know lipase they works on the lipids or fats, and the products we would be getting out of it would be the fatty acid and the cholesterol. Now we have to identify the organ that would be producing the lipase. So they have already given you the number of the organ, which was 4. So we identified it a while ago that this 4 is the pancreas with us. For the last, they gave you the name of the enzyme only, which is maltase. And the maltase, they are available within the epithelial cells or the lining of the small intestine, and this is the part of the small intestine, like we have to be considering this, like if we become specific, so you can write it as duodenum or jejunum, but generally they are the small intestine. The latter would be 5. The maltase we know works on the maltose, and we would get the glucose out of it. Moving onward, for the B part, they want us to be telling them the role of the hydrochloric acid that would be contributed by it in the alimentary canal. And we know that this hydrochloric acid, this is released into the stomach, and the glands that would release them, they are the gastric glands. The role of the hydrochloric acid is that in our food, if there are some harmful microbes, it would kill it up. The pepsin enzyme that works in the stomach, it requires the acidic pH for the optimum catalytic activity. So this acidic pH again is provided by the hydrochloric acid. So here 3 points we are done with. As of other point, you would say that since from the mouth, the food that has come via esophagus, there would be presence of the salivary amylase there in the food. So this HCl or the hydrochloric acid would denature it. For another 1 mark, they want us to state a function of the region of the alimentary canal, which is labeled 6. So the labeled 6, this was the colon with us, and the role of the colon is that it would absorb the water and the ions. The other approach can also be that the food that remains undigested within the alimentary canal, so once it comes here in the colon, so this is hardened, like the water is absorbed out of it, so it would harden and it would be becoming solidified and would take the form of feces. If I were to come here to the D part, and the total marks given were 6, and the examiner has to ask is that describe how the food which is eaten by the humans is reduced to smaller pieces. Okay, and explain how this makes chemical digestion more efficient. Now, here there is a link to the physical and the chemical digestion. Like the food that we have eaten Once this would enter into the mouth, so into the mouth, there would be happening of the physical digestion with the help of the teeth, like the incisors, the canines, the molars, and the premolars. The incisors, we know that they cut down the food. The canines that we have got, they tear the food. The premolars and the molars, they crush the food. So once that would happen, so the surface area of the food particle would be increased. And once the surface area of the food particles is increased, then it would make easier for the enzymes, like I was talking all of it with the context to the mouth. So it would make it easier for the amylase enzyme to be effectively binding with them. The other approaches could be that not only the physical digestion happens in the mouth, this also happens in the stomach. But in the stomach, we since cannot talk about the teeth, but rather than we would say that the stomach has got the muscular walls in it, and those muscular walls they help to churn the food, and again that would be breaking down the food particles into the smaller one, increasing their surface area for the effective digestion by the pepsin enzyme then. Now, here there is another way around in terms of physical digestion that the bile that comes from the gallbladder into the small intestine, although appears to be a chemical excretion, but it does not have any enzymes in them. So it again is considered in terms of the physical digestion, where it would emulsify the fat breaking them down into the small fat globules, increasing their surface area, and later would make it easier for the lipases enzymes to work effectively on them. So the same question I have explained three different approaches to it in terms of mouth, in terms of stomach, and in terms of the bile. Only the first scenario would change, like in the mouth related to the teeth, in the stomach related to the presence of muscles and bile with reference to or in context to the emulsification process, and the other points they would be the same. So if I were to read my answer for you, so we would simply say that the food which is eaten by the human, it is reduced to the smaller pieces by the mechanical or physical digestion, and this is done by the teeth in the mouth. Then I have said that the incisors, the canine, they cut the food. The premolars and the molar teeth, they grind the food or crush the food. And in that way, the surface area of the food particles is increased, not reduced, but rather than once they are converted into the smaller pieces, the surface area is increased. And this would make it easier for the enzymes like amylase, since I'm talking in context to the mouth, to be binding easily or effectively with them. Coming here to October, November 2022, the theory paper variant was 4.3, and for your exam, this was question number 2, but its part A and C, they were associated with the nutrition in humans chapter. What they have to be telling us is that the digestive enzymes are excreted into the alimentary canal, and we know why they are being excreted so that they can break down the large complex food substances into the smaller ones. What they further tell us is that as well as enzymes, other substances that are important for the digestion are also excreted into the alimentary canal. So not only the enzymes, but some other substances they are also secreted along with them, and for 2 marks they want you to state the name of those two other substances. So we know in the stomach there is excretion of the hydrochloric acid. This is not the enzyme. From the liver there would be coming up of the bile that temporarily gets stored within the gallbladder. So this again is the other excretion. Likewise, if we talk about the mucus, and we also have an example of the saliva, I have written 4, you were required to be writing any 2 out of it. Now, if we go on toward here, so this again is a kind of a question where we are required to be telling our examiner the name of the enzyme, the organ where the enzymes would is secreted, the organ when the enzymes acts. And at then you see that they ask you to be writing the product of the digestion when this would be catalyzed by the enzyme. For the first, they gave us the organ where the enzyme is secreted. This is the salivary gland, and we know in the salivary gland, the enzyme that we have got. So this is a salivary amylase, right? Not the saliva, but rather than the name of the enzyme would be salivary amylase enzyme, and where this enzyme acts, so this would be carried with the help of the salivary duct to be coming in the mouth, and it would act on the starch, and the end product we would obtain with the mouth is the maltose with us. The other name that they gave us was the stomach, and where this is secreted, and that is the stomach, and this also acts in the lumen of the stomach. And since it acts on the protein, so the final product we be getting out of it would be the amino acid. Now here they gave us the name where the enzyme is secreted is the pancreas. So we know that the pancreas, they release many enzymes like the lipases, the trypsin, and the pancreatic amylases. But considering the product of the digestion given here are the fatty acid and glycerol, so that is why the name we be writing would be the lipase. And we know that this lipase would be coming to the small intestine where it would break down the lipids to the fatty acids and glycerol. Other than that, they say that we have got this organ which is the small intestine, and where this is acting in the epithelial lining of the small intestine, and we have been doing this from the very start, and we know that in the epithelial lining of the small intestine, the enzyme that we have with us is the maltase, and the end product that we would be obtaining, this would be glucose with us. So this is how you would have scored total of 4 marks for this. If I were to move to the C part, what they have to be telling us is that lactase is another digestive enzyme and it is produced by the human babies. Now, for 2 marks, they want us to explain that why lactase is important for the nutrition of the young babies. What we know is that the young babies they are rapidly growing, so they need to release a lot of energy, and for the energy production, the substrate that is required is the glucose. Now, lactose that we give to the young babies in the form of the milk, that is a double sugar and has got the glucose and lactose linked together. So that is why what we would be telling our examiner is that the young babies have their nutrition and that is entirely based on the milk, and that milk has got this lactose sugar in it and it has to be broken down to glucose and lactose. So that is why they need to have this lactase enzyme so they can bring about the breakdown of the lactose which is present within the milk. For the C second part, what they have to be telling us is that many people they stop producing lactase when they become the adults, and if they drink the milk, this may give them diarrhea. No, the idea is very simple. Like if the lactase has got stocked in their bodies. So if they would be drinking the milk having the lactose in it, so it would not be broken down, and ultimately the unbroken lactose, when it goes to the large intestine, so it produces the methane, carbon dioxide, and the other gases, and it would give them the diarrhea and the gas. But they wanted us to be describing the symptom of the diarrhea, so we know that this would be the loss of the water effuses. If I were to come here in context of this diarrhea and they say that the cholera may be the cause of it, they want us to tell him that how come the cholera bacteria causes the diarrhea. Now you have to be understanding as that we have got the villi, okay, the villus, and in the villus what we have got are its epithelial cells, and these epithelial cells we know they have got the microvilli in them. When the cholera bacteria would come, so it would release a toxins, and that toxin it would cause these epithelial cells to bring about the excretion of the chloride ions into the intestinal lumen. And when the chloride ions would be accumulating in the intestinal lumen, so they would lower the water potential here, and automatically in these cells the water potential would be increasing, so water would move out of these cells and coming into the intestinal lumen down the water potential gradient with the help of osmosis, and this would be causing the diarrhea in them. So for 3 marks, the answer would be that we have got this cholera bacteria. This produces a toxin. This toxin causes the epithelial cells of the small intestine to release the chloride ions into the lumen of the small intestine. Due to the accumulation of the chloride ion, there would be a decrease in the water potential in the intestinal lumen, and this would cause the water to move out of the cells lining the small intestine to be getting out by osmosis, and this would lead to the development of the diarrhea in the individuals, those who are suffering from an infestation by the cholera bacteria. If we come here, so this happened to appear in the February March of 2023, and the theory paper variant was 4.2, and we would be doing its question number 2 of the exam, but only its part B and C. And this again is in context of the enzymes that contribute their role in the alimentary canal. So again, the table is same. We have the enzyme here, we have the substrate, and we have got the product or the products. They gave us the name of the pepsin, and we know the pepsin acts on the protein, and the product we would be making out of it is the amino acid. They gave us this end product, which was fatty acid and glycerols. So the enzyme working would be the lipase enzyme, and the substrate on which it would work, they would be the lipids with us. The trypsin we know is released from the pancreas. This comes in the small intestine with the help in the being present in the pancreatic juice. It would act on the protein, and we would have the amino acids out of it. At the end, they gave us the end product, which is glucose. So the glucose be made from the maltose, and the enzyme acting on it would be the maltase. And when the maltase would break down the maltose, so we would get two molecules of the glucose, and they already wrote the glucose here. So we would be writing maltose here and the maltase enzyme here. If we were to be talking about the C part, they want us to name the structure that increases the surface area for the absorption in the small intestine. So you have the choice of either writing it as the villi or the microvilli. Moving onward to this question from May June 2023, the theory paper variant was 4.1, and for your exam, this was question number 1 with us. And here again, we have to be identifying the structures of the alimentary canal and what role they would have in the digestive process. What I see is that A, they are the salivary gland. The L that we have got, so this would be the mouth. The B is the esophagus. The C that we have here is the stomach. D is the pancreas. K that we have here, this is the liver. And this guy right here, indicated with the letter J, so this is the gallbladder. And this part that is indicated with H, so generally they are the small intestines. The E part that we have here, so this generally is large intestine, but if we become specific, so this is the end part of the colon. And the G that we have here is the rectum, and the F that we have got at the end, so that would be the anus with us. Now let us see what the examiner has to be asking was that each letter that we have given you above in the diagram, it can be used once, more than once, or not at all. We are required to be stating the latter that would relate to the function, and they say that produces bile. So the bile production is done by the liver. So the latter we would be writing here, it would be K with us. That produces gastric juice. So wherever the word gastric comes, so this means this is related to the stomach, and the latter that we have here, this is C, so this would be the stomach. But since we have to be writing the latter, so I'm not writing the name of the organ but the latter. And they say that produces urea. So urea, although is excreted in the urine, but this would be produced within the liver, which is the letter K, by the deamination process. So here the K comes again, where the maltose is digested. So the maltose would be digested within the small intestine, where it would act on the maltose disaccharide and would convert it into two molecules of the glucose. So we are writing letter H here. And they say where the trypsin acts. Trypsin is a protease enzyme, although is produced by the pancreas, but it would be brought to the small intestine with the help of the pancreatic duct. So again, the trypsin would be acting within the small intestine, and we would be writing the letter H for it. Now coming to word, a question from May June 2023. The theory paper variant was 4.2, and this was question number 7 with us. The examiner wanted us to complete the sentences, and this again was about the enzymes, and where we are required to be writing a suitable word or the phrase in each of the space provided. So how you are required to be cracking is that you have to read the complete statement and then you have to be writing a suitable word or a phrase in the blanks. What they wrote was that enzymes are involved in the chemical digestion which produces small dash molecules that can be absorbed into the blood. Now, since the reference says absorbed into the blood, so these small molecules they need to be soluble. Later they say the two examples of the protease enzymes are the pepsin and trypsin. Pepsin is produced by the dash and requires acidic condition, and we know it very well that there is pepsin and it requires acidic condition. So where this is produced, it is produced in the stomach. Later they say is that these conditions are created by the release of dash, which provides the optimum pH for the pepsin activity and also kills harmful what? That would be the microbes. And we are aware that within the stomach, the acidic conditions, they are basically provided by the hydrochloric acid. If we move further, they say the dash produces trypsin, which breaks down protein in the dash pH conditions. So we know the trypsin, it is produced by the pancreas. And if they have to be breaking down the protein, so there would be a need of the alkaline pH conditions. They say these conditions are created by a substance called dash and which neutralizes the gastric juice. Now, although the trypsin is acting in the small intestine, and even though this is requiring the alkaline pH conditions, so this would be provided by the bile because the food that arrives from the stomach in the form of the chyme, so that has still got some gastric juices in it, so the bile has to be available. And then they say it also has an important role in the dash of fats and oils. Now we know that for the fats and oils there is need of the emulsification. So by just writing, by just reading the entire statement, we were able to crack the word bile here and emulsification here. Moving onward, here we come to May June 2023. The theory paper variant was 4.3. And for your exam, this was question number 7, A and B part with us. What they have to be telling us is that state the name of a chemical element that is found in all proteins, but not in carbohydrate or fats. The carbohydrate and the fats they are made up of carbon, hydrogen, and oxygen, but they never have the nitrogen in them. So the answer becomes simple. We would be writing nitrogen here. The B part is related to the enzymes that are involved in the digestive process. We were required to fill up the table where initially they gave us the name of the large biological molecule. The breakdown product that we would have when the large biological molecule would be broken down. The enzyme that would bring about the breakdown of the large biological molecule and the organ that produces the enzyme. Now, since the first one is the oil, so the breakdown product would be the fatty acid and glycerol. The enzyme working would be the lipase enzyme. Organ, they themselves gave the name, and this is the pancreas. The glycogen we know is a complex sugar, and they gave us the name of the enzyme that this glycogen would be broken down by the glycogen phosphorylase enzyme. The organ where the enzyme would be produced, this is the liver. So if glycogen has to be broken down, so the breakdown product that we would obtain, this is glucose with us. Likewise, they gave us the name of the product of the breakdown. This is maltose enzyme was amylase. So we are aware of this fact that this would be the large biological molecule from where the maltose can be obtained. So this is starch. And the breakdown of the starch, we are aware that this is brought about by the amylase, but the amylase is being produced by two organs. One of that is the salivary gland, the other one is the pancreas. So this is up to your choice. You write either this or this. At the end, they gave us the name of the large biological molecule, which is protein. So we would be getting amino acids out of it. The enzyme working would be the protease enzyme, and we know the organ where this would be produced. This is the stomach with us.
Moving onward, and here we come to October November of 2023. The theory paper variant was 4-1, and for your exam, this was question number 2 with us. And mostly this is focused on the structure of the villi that we have in the small intestines. What the examiner has to be telling us is that the small intestines, this is lined with many villi and is adapted to absorb the glucose. They have given us figure 2.1, and this shows two villi from the lining of the small intestine, and these villi actually makes the internal part of the small intestine. And if you were to see clearly, this is the one villus that is being shown, and this is the second villus that is being shown. If we just come at the center of these villi, so what we have here, these are the lacteals, a specialized kind of the lymphatic vessels that actually absorbs the digested products of the fats, which is the fatty acid and the glycerol. And he would give you a hint with regard to M. So this cell here that I am marking for you as of now, this is a goblet cell, and the role of this is to be producing a slimy excretion which is mucus with us. For 1 mark, they want you to state the name of the structure in, and we have got it at the center, so this would be the lacteal with us. They tell you is that cell M that we show you in figure 2.1, this creates the mucus. So the mucus creating cell, we know this is the goblet cell with us. For another 1 mark, the examiner wants us to state another place in the body where the mucus secreting cells are found. So not only that we have these goblet cells secreting the mucus in the small intestines, there are other parts or the other organs in our body where the mucus would be secreted, and that includes the trachea that we have as a part of our respiratory system. You can write the bronchi and as well as a bronchiole. So any one word from here that where the mucus cell would also be found. For another 4 mark, the examiner wants us to describe and explain how the structure of the villus is adapted for the function. And what we know is that the major role of the villus is to be absorbing the digested food particles and then taking them into the blood capillaries and as well as into the lacteals, which are the specialized type of the lymphatic vessels with us. So I am just quickly going to be making the structure of the villus to be telling you about the four major adaptations that are related with respect to the villus. So the first point would be that in these villus, what we have are the epithelial cells. These epithelial cells, they have the microvilli, and these microvilli, they would be increasing the surface area to the volume ratio, and ultimately this would be resulting for the most efficient absorption. Beneath these epithelial cells, what we have got are the blood capillaries, and these blood capillaries, it has a good blood supply. So the blood would continuously enter into them and would exit through them, and in doing so, it would help to maintain the concentration gradient for the movement of the digested food particles that lie here outside of the villi in the lumen of the small intestine and within these blood capillaries. So here we have just covered 4 points like the microvilli having its association with an increase in the surface area to volume ratio, and then the blood capillaries having the good blood supply to be maintaining the concentration gradient difference. So these are 2 points each. If you were to be writing some other points, so you can talk about the lacteals that would absorb the fatty acid and the glycerol. And the other important point as an alternative valid point would be that these epithelial cells or the villi overall, they have the thin walls, so that would reduce the distance over which the diffusion would take place. If I were to go toward the B part, they want us to be naming the main blood vessel that would take the blood from the small intestines and the liver. So if I have got these small intestines, and then here we have got the liver. So if I am taking the materials, or I am taking the absorbed food materials from the small intestines to the liver, so here the main blood vessel that would be available.
Which is hepatic portal vein with us. this right here. If I were to be coming to the C part, what they have to be saying is that all cells they develop from stem cells describe what is meant by the term stem cells. In our body, there is presence of the specialized cells like the neurons and the red blood cells and the ciliated cells. So all these specialized cells, they basically come from the unspecialized cells and they are the stem cells. And the scenario with the stem cells is that they have an ability to divide by the mitosis process. And when they would do so, so they would be making the new daughter cells, and these new daughter cells later they develop some unique structures, and that would allow them to be becoming a specialized cell or a differentiated cell. So for 2 mark, we would simply say that they are unspecialized cells. They divide by the mitosis, and as a result of that, they produce new daughter cells. If there was a 3rd mark, so you would have added a point that these new daughter cells, they develop the new structures that make them specialized for a specific role. If I were to come toward the D part, and here they say is that some cells that we have got in the small intestines, they have many mitochondria. And we have been doing many of the past paper questions, and these are the epithelial cells that we have within the small intestines to be possessing a lot of mitochondria in them. In the graph below, they have given us the average number of the mitochondria that are found in 3 different cell types. Like we have got the heart muscles, then we have the mature red blood cells, and then we have got the epithelium in the small intestine. Now, this is something presented on the x-axis. And if we were to come on toward the y-axis, so here we have got the average number of the mitochondria per cell. And if I were to simply see, so comparative to the mature red blood cells, the heart muscles, and the epithelium that we have in the small intestines, in them the number of the mitochondria is far more greater, and I don't see any of the mitochondria within the red blood cell. Now, if we move to the question, so here the question was in terms of the total of 5 marks, and they wanted you to explain the differences in the average number of the mitochondria in each type of the cell. Like we have the heart cell, we have the epithelium in the small intestine, and we have got the red blood cell. So we need to be explaining the differences, not the description. So if I were to be quoting you some of the points, so what we know is that these red blood cells that we have, so they need to have more space for the packaging of the hemoglobin protein. So that is why out of the mature red blood cells, the mitochondria are removed. The other point is that these mature red blood cells, their role is to be binding with the oxygen gas and to be releasing the oxygen gas at the point of the respiring tissues. And at binding with the O2 and releasing the oxygen gas, this is by the diffusion process. So there was no need of the mitochondria for these red blood cells. If I am to be explaining it in context of the heart muscle, so the heart muscles they keep on contracting from the age of 2 months till we die. So the heart muscles they are contracting to be pumping the blood around the body. So for their muscular contractions, there is need of such an immense number of the mitochondria per cell, and in this case this is 1500, but we would not quote 1500 because the question is in terms of explanation. If I was to talk in context of the epithelium that we have in the small intestine, so again in them, the number of the mitochondria, but we see from the graph is the highest, and the reason is that these epithelium in the villus of the small intestine, they absorb the digested food materials with the help of the active transport. And without the active transport, the digested food materials cannot be absorbed. So that is why these epithelium needs to have a higher number of the mitochondria in them. So if I were to take you or to be reading my answer as per the mark scheme or the examiner report, so you just have to be focusing as that I have written that mitochondria they are the site of aerobic respiration, where the glucose would react with the oxygen gas to produce energy. In the red blood cells, there is need of more space for us to be packaging more of the hemoglobin so that they can bind and release. And the other point is that they bind and release oxygen gas in red blood cells with the help of the diffusion process. So there is no requirement of the mitochondria in them, right? And the heart muscles, since they contract continuously, So they need to have abundant mitochondria to release energy for this. The epithelium in the small intestine, since they absorb the digested food particles with the help of the active transport, and for that purpose, abundant mitochondria has to be available, so they release energy for the active transport process to be occurring. Moving onward, and here we come to the May June of 2024. The theory paper variant was 4.2, and for your exam, the question number 4C second part and question number 6 was from this chapter. Let us just see what the examiner has to be asking us, that describe how the starch is broken down into the human digestive system so it can be absorbed into the blood. So I have eaten the starch and then I need to be breaking it down within my digestive system. So finally it can be converted into the glucose molecules where we can absorb it into our blood plasma. So the marks given were 6, and I would be explaining you with the help of the rough diagram, and then we would come to my answer. So what we believe is that we have got in our alimentary canal this mouth, and this is the site or the place where the digestion of the starch actually begins. And the thing happening here is that when the starch we have got it into the mouth, so two mechanisms would be taking place, or two type of the digestions would be occurring. We will have the physical digestion taking place here with the help of the teeth, where the teeth would cut the food into the small pieces, and that would increase the surface area. And at the same time, what we believe is that we have got the salivary glands and they would be sending the saliva into the mouth. And that saliva has got an enzyme in it. And the name of the enzyme is the salivary amylase enzyme. And this salivary amylase enzyme, it would be acting on the starch and it would partially break it down into the maltose molecule. Not all the starch is converted into maltose, but some of it would be converted into the maltose and some would remain undigested. In the mouth, this food would be converted into a spherical mass, and that we know is the bolus. And this bolus would then come into the esophagus, where the peristalsis would happen. And finally, this starch and the maltose, they would come within the stomach. In the stomach, we do not have any enzyme to be working on the leftover starch or the maltose, but what we know is that the stomach has got the muscular walls, and these muscular walls they churn the food, and that churning is equivalent to the physical digestion that would be happening here. Afterward, this would be coming into the small intestines, and in the small intestine, two things can happen with context to the starch digestion. The first way is that we have got these pancreas. The pancreas would be sending the pancreatic juice, and that pancreatic juice it would have the pancreatic amylase in it, not the salivary amylase, but rather than we have got the pancreatic amylase coming here, and this pancreatic amylase this would be acting on the leftover starch that we have brought from the mouth and then esophagus and stomach, and finally this is coming here in the small intestine. This would be converted into the maltose. Maltose still is a disaccharide, a double sugar that cannot be absorbed by the blood capillaries. So what we believe is that within the small intestine we have got their villus, and in the villus we have the epithelium cell or the epithelial cells, and in the epithelial cells we have got the maltase enzyme. This maltose would bind with the maltase enzyme, and finally this would be broken down into the glucose. Now, this is something that is small enough or is a simple sugar that can be absorbed into the blood capillaries. So, this is the overall context of the answer that we were required to be writing here. So, if I were to simply read my answer, that would be that in the mouth, the starch that we have got, this is broken down into the small particles, and that would be equivalent to the mechanical breakdown by the teeth. At the same time, within the mouth, the salivary gland, it would release the saliva and it would have the salivary amylase in it. The salivary amylase would break down the starch into the maltose, and then the food is converted into the bolus, and finally it would be brought to the stomach via esophagus. In the stomach, we have the muscular walls that would churn and break down the food physically. It would be then moved to the small intestine, where the leftover starch is broken down into the maltose by the pancreatic amylase. So here the 6 points are done. But I have written the last point with context to an extra knowledge that we have got these epithelial cells within the villi where there is presence of the maltase enzyme, and that would be acting on the maltose and would break it down into the glucose. Now, glucose is something that we have got from the starch and we can take it into the blood. So the same paper, and if we come toward question number 6, so it was relative to the functionality of the teeth and the villus. What they have to be saying is that we are required to be completing the sentence that is relative to the human teeth. They say the process of taking food into the mouth is called dash, and we know this is ingestion with us. The flat, sharp edged teeth that we have at the front of the mouth, now we know that they are the incisors, like they are the flat and they are sharp edged teeth, and they are used for biting off the pieces of the food. And then they say the dash and dash are the large teeth toward the back of the mouth. So at the back of the mouth, the large teeth, they are the molars and the premolars with us. And these broader teeth, we know they can be used for dash the food. So you can use any of the word grinding, crushing, or chewing. If we were to move to this B part and they show us the diagram of the villus, I will not be explaining any of it as of now. Let us see what they have to be asking. And they say they draw labels and label lines on figure 6.1 to identify a capillary and a lacteal. So we have done like a lot of questions from this. So at this point, I hope you would be very much clear that these are the blood capillaries. At the center, we have the lacteals. And here we have got this epithelial layer, and each of the cell would be the epithelial cell. So I would go from here, and this would be capillary. I would go from here, so this is the lacteal with us. They happen to be asking us that state the function of the lacteal. So we know they transport, or either you would say they absorb the fatty acid and cholesterol. For the last C part, they want you to describe the pathway that would be taken by the product of protein digestion from the villi to the liver. Now, the product of the protein digestion, we know this is the amino acid. This amino acid initially, what this is going to be doing is that it would pass through the epithelial cells and then it would go into the blood capillaries that we have in the villi. From the blood capillaries, this would go into the hepatic portal vein. And from the hepatic portal vein, we would take it to the liver that we have in our body. So, this is the pathway that would be taken from the product of the protein digestion all the way from the villi and then ultimately coming into the liver. So, how the answer would look like that the amino acid which are product of protein digestion, they firstly passes into the blood capillaries and then they are taken to the liver with the help of the blood plasma. So this is how you would have scored 2 marks for this. If I were to come here, and this is from October, November 2024, the theory paper variant was 4-3, and this was question number 1 for your exam. A kind of a mixed question from the identification of the parts of the alimentary canal in terms of bile and the breakdown of the protein by the trypsin. They tell us says that we gave you this figure 1.1 and this shows you the part of the human digestive system. And they want us to be using the label lines and the latter to identify on this figure the organ that produces bile and label it as A. So we know that the bile is produced by the liver, so we would go from here and we would mark it as A. And the organ that stores the bile and label it as B. So here we have got the gallbladder, and this gallbladder is what that would be storing the bile in it. Moving to the second part, for 2 marks, they want us to describe the physical digestion. So we are aware of this that the physical digestion that would break down the large food particles into the smaller pieces, and it would not do any of the chemical change to the food. For the third, they want us to state the name of the two places where the physical digestion occurs in the body. So they are the prominent one, the mouth where we have the teeth, and in the stomach we have got its muscular wall. Coming toward the B, this is from the bile aspect, and they say that bile is involved in the fat digestion, not that the bile would bring the chemical digestion, it would bring the physical digestion, and it breaks down the large fat molecules into the small fat globules, and that process goes with the name of emulsification. Once the emulsification is done and the large fat molecules they are broken down into the small fat droplets, then it becomes easier for the lipases enzymes that come from the pancreas into the small intestines to be effectively breaking them down. So we need to be completing the sentences that are relative to the fat digestion. They say the bile is released into the dash in the digestive system, so this is released into the small intestine. Further, they say the bile increases the surface area of the fats and oil, and we know this process would be referred to as emulsification. Later, they tell us is that this increase in the surface area, this would increase the rate of chemical digestion. And using the enzyme word, that is a lipase for us. For 1 mark, they want us to state the names of products. See, the word is products of the fat digestion. So from the fat, we would get fatty acids, that would be 3, and we would be getting 1 glycerol out of it. For the last C part, they want us to explain the role of bile in the breakdown of the protein by the trypsin. Now, the trypsin enzyme is working in the small intestine, and this has to break down the proteins into the amino acid or the peptides into the amino acid. But this trypsin enzymes requires an alkaline pH. The food that comes from the stomach into the small intestine, that is in the form of a liquefied food that is chyme and that is acidic in nature, having the hydrochloric acid in it. So when the bile would come into the small intestine, what this is going to do, it would neutralize the acidic mixture that has come from the stomach. And by that, it would provide the alkaline pH, and that alkaline pH would allow for the optimum activity of the trypsin enzyme. If we come here to the February March of 2025, the theory paper variant was 4.2. And for your exam, this was question number 3 with us. Related to the water and the cholera bacteria that causes a diarrhea in the alimentary canal. They have given us this figure 3.1, and this shows us the percentage of the water that we have got within different structures of the human body. Like the brain has 73% water in it, the blood 94%, the kidneys have 79% water in it, the muscles have got 75% water in it, and so are the other organs, and their water content is written right in front of it. Now, this is a little numerical, and they say the mean mass of the human eye is 28 grams. So, they are giving us the total mass of the eye. For the human, this is 28 grams. They want us to use information from figure 3.1, where we are required to calculate the mass of water in the human eye. Like the overall mass of the eye is 28 grams. So, they want us to calculate at how much mass of water there is in this 28 grams. And the answer should be up to two significant figures. If you would not be doing it, you would lose 1 mark. What they have given us is that the percentage of the water in the eye, this is 95%. So I would see that how much 95% would be of 28 grams. So here this is 95, I would divide it by 100, then I would multiply it with the total mass of the water in the eye. This is 28 grams. So the answer I got was 26.6 grams. So since we have to be writing near to the two significant figures. So these are two significant figures, but after the point, this is beyond zero point 0.5. So I would round it off to 27 grams, and here we have got the final answer. Any of the student who would have written 26.6 grams, they would only get 1 mark. They want us to be describing the importance of the water in the human body for the second part against 3 marks. So the idea is that this water that we have in the body, this is a universal solvent, like it would absorb the nutrients and the ions and the hormones in it. What we know in terms of the elementary canal is that we have got the enzymes breaking down the large food particles into the smaller ones, and these enzymes they are hydrolytic in nature. And by the hydrolytic, what we mean is that these enzymes can only break down the complex food particles into the smaller ones in the presence of water. Other than that, what we believe is that when we sweat, so the water would come on the surface of the body, and here when this sweat having the water would evaporate, so this would help to be cooling down the body. The other roles you can be talking in context of the excretion as well. Here we come to the B part, And they tell us is that the cholera is a disease which is caused by the pathogen that is available within the contaminated water. They give us figure 3.2, and that is a diagram of the cholera pathogen. And for the first part, they want us to state the name of the structure L in M that is shown in figure 3.2. So we know it very well that this outside of the cholera would be the cell wall, and this L would be its cell membrane. They themselves have labeled this DNA and the plasmid, but they want us to be identifying M. So these M that we have got within the cytoplasm of the bacteria. So these are the ribosomes with us. For the second, they want us to identify two features that are typical of the prokaryotes. So the typical of the prokaryotes would be their circular DNA that we have here, the presence of the plasmids that they have in them. Okay, so this would be the circular DNA and the plasmid, and we can never forget about the cell wall. You can be writing it as an alternative point. For the third point, they want you to be stating the type of the pathogen that causes cholera. So the type of the pathogens can be the bacteria or the virus or the protozoa or the stuff like that. And we believe from our theoretical knowledge that this cholera, with context to the type of the pathogen, is bacteria. For the 1 mark, they have given you the scientific name of this, and we know that the first part of the scientific name is the genus and the second part is its species. So since they have asked us to be stating the name of the genus, so we would be writing the Vibrio here. If I were to come toward this point, for 3 marks, they want us to explain that how the cholera pathogen would cause dehydration in the human body. So the cholera would come, it would release a toxin here. And when the toxin would be released out of the cholera, so it would cause epithelial cells to be pumping the chloride ions out. And when the chloride ions they are pumped out of the epithelial cells, so they would reduce the water potential here on the outside of them. And the water potential within the epithelial cells, this would increase. And with the help of the osmosis, the water would go out of the epithelial cells, coming into the lumen of the small intestine. And by that, we would be developing the diarrhea. So, as written for the answer is that it produces toxin. This toxin would compel the epithelial cells of the small intestine to release the chloride ions, and that chloride ions have come into the lumen of the small intestine. When there is presence of the chloride ion, this lowers the water potential here, and then it would cause the water to move out of the epithelial cells by osmosis, and that would lead to the diarrhea. Moving toward the last question from this chapter appeared to be in the May-June of 2025. The theory paper variant for your IGCSE Biology was 4.3. This question was relative to identifying the parts of the alimentary canal and some questions related to the enzymatic activity that goes on here within all of it. So I would not identify the parts here. Let us just simply go to the question that what they have to say, and then we would crack the diagram. They say that table 1.1 it shows information about some of the structures which are shown in figure 1.1. We are required to be completing table 1.1, and for that we need to use figure 1.1. So for the first they have given you the letter G. So we would come here and see what the letter G is. So this is the anus with us. Okay, why I didn't explain the diagram, I hope you may have got an idea. We need to go to the question first. So this G that we have got, this is the anus, and the role of the anus would be that it would allow the removal of feces or undigested food particles. So the answer is simple, either you would say egestion or the removal of feces. They have written produces amylase. So the amylase production happens here within the salivary gland, that is the M with us, and that would have the salivary amylase. The amylase also is released by the pancreas, and that would be the pancreatic amylase. So you have the choice of either writing the letter M or the D. But if you're writing M, you would have to be telling him that this is salivary gland. And if you're writing D, so you would say that these are the pancreas with us. So simply the amylase production is either from the salivary gland, the latter would be M, or it is from the pancreas and the latter would be D with us. They say gallbladder. So we need to identify the gallbladder. The gallbladder would always be present with the liver. So here we have this liver, and with the liver lies this L structure. So this is the gallbladder, and the role of the gallbladder would be that it stores or it releases the bile. They say transport food to the stomach. So the transportation of the food to the stomach, this would be done with the help of the esophagus, and the latter would be B with us. So this is how you would have cracked total of 4 marks. If I were to come here for the B1 and they want us to describe the role of the chemical digestion, the marks were 3. So you just need to follow me that in the chemical digestion, the large food particles that we have caught, they would be broken down into the small food particles chemically. And by the chemically, we mean are the enzymes, and the food would be chemically changed. Now, after they have got chemically changed into the smaller particles, so for scoring the last mark, we would say that they become small enough that they would be able to be absorbed into the blood. So the first mark would be that it is a breakdown of the large food particles into the small pieces. And the second point would be that in doing so, the food particles, they are chemically changed. And the third point would be that they become the soluble molecules, so we can take them into the blood. The second point is that the maltose is digested by the maltase, and they want you to name the product of this reaction. So the maltase, we know that this would be acting on toward the maltose, and as a result of that, the product of the digestion we be obtaining that would be the glucose with us. For this point, they want us to tell him that where the maltose is digested in the small intestine. So, in the villi, we have the epithelial cells, and in the epithelial cells, we have got this maltase enzyme. So, the answer would be simply that it would be occurring in the epithelial cells of the small intestines. If we were to come to this fourth part of the question, and for the total of 6 marks, this was a little technical but was very easy. They asked was that why the amylase digest starch in the small intestine but does not digest starch in the stomach. So there were two approaches to the answer. Now, what we know is that we have got these small intestines, right? And in the small intestines, if we were to be digesting the starch, so there would be coming up of the enzyme for it, and that would be the pancreatic amylase. The pancreatic amylase, this basically prefers the alkaline pH. Now, if we talk with context to the stomach, so what we know is that in the stomach there is presence of the acidic pH, and that acidic pH is provided by the hydrochloric acid. So if I were to bring this salivary amylase enzyme here, so what would happen? That this hydrochloric acid, it would denature the salivary amylase. And when the salivary amylase is denatured, then as a result of this, this enzyme would lose the shape of its active site. And when the active site shape is lost, then it cannot be binding with the substrate molecule. And as a result, no enzyme substrate complexes would be formed. And ultimately, when no enzyme substrate complexes would be formed, then ultimately the starch cannot be broken down. So this was the first approach. The other approach could be that half you talk about that the hydrochloric acid since provides acidic pH, so the amylase enzyme would be denatured. And then you start talking about the small intestine, that in the small intestine we have got the alkaline pH that is provided by the bile. So that would allow the salivary amylase to be binding with the starch and to be making the ES complex, and finally we would have the product out of it. So either you restrict to the hydrochloric acid and you write all the points in the answer, or you write half in term of stomach and half in term of the optimum pH that is provided by the bile. So if I were to take you to my answer again 6 marks, so from here and till here, this is the first approach written, okay? And if you want to do the other way around, where the half point would come from the stomach, so they would be here, and the half would be in term of the bile scenario. So you just have to follow me. What I have written here is that since we have the stomach, and over there is presence of the hydrochloric acid, and this acid, it lowers the pH of the stomach, and in that lowering of the pH, it would denature the amylase enzyme. And when the amylase would be denatured, so it would lose the shape of its active site, and it would no longer remain complementary to the substrate molecule. And as a consequence of that, no enzyme substrate complexes would be formed, and the starch would not be digested. So here we complete 6 points, just sticking to the stomach scenario. The other way around is that half you must write from here to here, and then the next point would be that in the small intestine the bile is released. And from where this is coming? From the gallbladder. And this bile, since in the small intestine, would provide the alkaline pH and it would be neutralizing the acidic mixture that is coming from the stomach. So ultimately allowing the amylase to be digesting the starch. So I hope you would be very much clear with all the past paper dimensions for this challenging topic of IGCSE's biology that was entitled as a human nutrition, and I have done all the questions from this chapter from the year 2016 and 2025. If any of the year that I have missed, so this means that the question did not appear in that year. So see you in the next class where we would be doing the transport in humans.